Hello, hello everyone. So I just found out today that I'm the president of the jury. So I'll do my best to introduce the proceedings. So first of all, for the purposes of this defense, you are in the University of Strasbourg, just for the duration of the defense. And so today, we're going to have a presentation by the candidate, Theo Barracho, and well, you can see the thesis title up there, Demographic and Behavioral Responses of a Polar Species to Environmental Variability. And first of all, I'll just introduce myself and the other members of the jury. So I'm Richard Phillips. I'm from British Antarctic Survey, and I'm the head of the predators group there. And I work on albatrosses and petrels.
Hello, also from me, I am Sandra Bauhaus. I'm the scientific director of the Institute of Avian Research in the north of Germany, and I run a long-term study on common terns.
Hello, everyone. I am Amélie Lescroel. I'm a research associate at Point Bleu Conservation Science in California, and I've been working on Adélie penguins for the past 20 years.
And I'm Céline Boeck. I'm the PhD supervisor of Theo, one of the PhD supervisors of Theo. I'm the leader of the Polar Biology Department at the Scientific Center of Monaco, and I'm a researcher at the CNRS University of Montpellier. And I'm an ecologist, seabird ecologist, working on penguins mainly in subantarctic and Antarctic area. Maybe Jean-Yves Georges can present himself.
Bonjour, I'm Jean-Yves Georges. I'm a senior researcher at Institut Pluridisciplinaire Hubert Curien. I'm working on freshwater ecosystems, but I'm happy jumping in some salty water with Theo Defense. And thanks for inviting me as an examiner.
Hi, everyone. My name is Nicolas Leconte. I'm a professor in ecology at the Department of Biology at University of Moncton in New Brunswick, Canada. And I'm quite glad to have this defense going on today. And we have been working very hard to make this a very nice presentation and work. We are very also focused on understanding what are the changes in the ecosystem happening in polar areas in our lab, and the focus mostly on birds and their predators. And I wish I was there with you, but unfortunately I couldn't join you. So I hope we will have time through the questions and also after to interact as much as possible.
Okay, thank you. Well, and thank you very much for the other examiners introducing themselves. So the format is there'll be 45 minutes of talk from Theo, and then they'll move on to 30 minutes of questions from myself and Sandra as rapporteurs, and then 20 minutes of questions from the other jury members, and then 5 minutes from the supervisors. So over to Theo. Thank you.
Yeah, thanks to you. Well, thank you everyone for being here today. It really means a lot to me. And so, yeah, during this presentation, we're gonna talk about how Adélie penguins in Antarctica respond to variability in their environment and notably sea ice. But before we dive in a little bit more with the penguins, I couldn't resist to quote this sentence, which says, "Who could have predicted the climate crisis?" Well, it's a bit ironical, I have to see it this way. But yeah, do you know who said that? Yeah, well, hopefully not someone with responsibilities. But jokes aside, the reason I wanted to quote this is because it makes me think of a question we may not think about enough as a society.
Theo?
Yeah?
Excuse me, but we don't see your slides moving. We only have your first slide on our screens.
Okay. I think the régie is trying to fix this.
Thank you, the régie.
Or is it?
Prima
Okay.
Thank you.
Yeah, it's
Should work.
working.
So if I do the joke again, it's not gonna be funny, but okay. So yeah, just for Jean-Yves and Nico, I was saying, hopefully not someone with responsibilities. Anyway, so do you see the slides going, Nico and Jean-Yves?
Yeah, it's all good for us.
All right, great. So yeah, I was saying the question that this quote brings us to is why? Why do we need to predict? What does it bring to society to be able to predict what is going to happen? So having one supervisor from Brittany, where there are a lot of fishermen, and another supervisor from northeastern Canada, I thought I could give you the example, tell you about the example of the cod stocks in Newfoundland and Labrador. And what happens there, if you look at this time series of abundance, it's an index, but it's the idea, so the amount of cod in the area, you can see that in the 90s, there was a real population collapse. And the consequences of this collapse of the stock on the economical and social environment there were catastrophic because Newfoundland lost around 10% of its human population due to emigration after that crisis. But the main thing that is really interesting about this example is what happened actually in the decades before this collapse. And what was observable already as early as the 60s is that the size of the cod that were caught were smaller and smaller. And this conveyed the idea that there may be some signals, some hints that something is going to happen that we can observe before there is a collapse or before there is a strong change in population size. So what do we want? What is it that we want to predict when we research biodiversity in ecology? In our population ecology, more specifically, maybe it's usually the key metric is population size. So to tell you about this, I'm going to show you an example of black-browed albatrosses in South Georgia. So it's just a little population within a bigger one. Yeah, it's not chosen randomly. So this is a time series of population size. So how do you make that? You go at the place a few times, and every time you go, you just count the individuals or the pairs that are there. And what we are also very, very interested in with this metric of population size is to see how it changes through time. So for example, here it decreases, and this kind of information on the change in population size is what we use. We can use also other things, but that's one of the key metrics we can use to classify a species as threatened or not. So now we can observe that there is a change, but the thing we want to know is why. How is that changing? An approach to answer that question is to decompose. So we have here the first step is the population size. But the first decomposition of that we can make is that we can decompose it in values, in quantities that are called vital rates. So among these rates, you have the survival, either of the adults or the juveniles. You can have immigration and emigration, breeding propensity, so how many individuals attempt to breed. And you can also have reproductive success, for example. And if we keep decomposing that, behind every vital rate, there are actually individual traits that can be behavioral or physiological that are going to affect these vital rates. So for example, the physiological traits can be size or mass. For example, in fishes, the biggest fishes also make the most eggs, or it can also be behavioral. So for example, in seabirds and in birds in general, the duration of the foraging trips, for example. And if we keep going up the cascade, then we find the environment that is often responsible for this variability. And so here, for example, we have the increase in sea surface temperatures since the 1900s. So now you can start to see the idea of a cascade between the environment and population size. And to exemplify this a little bit more, I thought I could tell you about a real-life example, and actually one on yellow-bellied marmots in the US. So in this population, what the people that were working on them saw is that there was a huge increase in population size after the 2000. And they wondered, why is that? Why did the population increase? And at this point, it's anecdotal, but they used a model called an integral projection model to try and understand the cascade that was responsible for this increase. So the first thing they realized that this increase was driven by higher adult survival, that was itself driven by higher body mass before the winter season, that was itself driven by springs occurring earlier and earlier due to climate change. So now another question we may ask, because this is really interesting, this detailed knowledge, but it's super useful for understanding. But the question is, can we use it to improve our predictions, to improve our forecast? And when we say better forecast, better prediction, what does it mean? It means forecasts that are both timely, so at time scales that are interesting to us. So it can depend, it can be short or long, depending on the questions, and also reliable, so with as less uncertainty as possible. So the framework in which I wanted to tell you to present the things today is the one that is aiming at doing just that, using this detailed knowledge to try and predict. And it's a framework called the timeline to collapse. But we'll see that it can also be used for other things than just to predict collapse. So it comes, I think it would be interesting to do a bit of history. It comes from the early warning signal theory. So if you consider that this is a time series of abundance, population size, this theory says that you will have statistical signals, statistical artifacts before a decline that can be used as early warnings. But the timeline to collapse framework is a development of this theory that use multidimensional signals. So what do we mean by that? So it's really also something I should precise is that it's particularly focused on ramping stressors. So for things that stress to the species or individuals that are increasing through time. So for example, here, a decrease in food abundance. So the framework says that the first thing that is going to change in response to the decline in food abundance is going to be either the behavioral or the physiological trait. So for example, here, an increase in trip duration. Then we will see a signal in the vital rate, so for example, the reproductive success, and then only ultimately at the end, we will see an effect on the population size. And this framework really emphasizes the idea of looking at this sequence of consequences on different values, different metrics. And so, yeah, we were saying why this framework? Well, the first reason is that it can be useful. 50% of the reason is that it can be useful for predicting decreases mostly, but also increases. But also, and that is really important, I'd like to point your attention to that, is that it's very useful visually to understand the links between environment and changes in population size. So what do we have of validation? It's quite a recent, I mean, the ideas behind the framework are not recent, but the framework itself is quite recent. And we have partial laboratory validation of this framework on paramecium in microcosm. So here they exposed the paramecium to a stress at H equals 0 hours, and they observed a decline in population size at 111 hours. And what they were able to show is that the change in behavior, so it's the swim speed of the paramecium, that started to be informative about the decline in population size at 73 hours. So compared to the lifespan of those organisms, it's quite an early signal. And they didn't test the vital rates. That's why it's a bit still in validation. But the question we want to ask today is, can we also apply this interesting framework to wild populations? So for example, in the case of today, Adelie penguins in Antarctica. So you may think, if you know a bit about Adelie penguins and other seabirds, you would know that they are long-lived. So that means they, for example, in the case of the Adelie penguin, they can live up to more than 20 years. So that's not really practical for studying these things, especially with the fact that they breed in Antarctica, which is not really accessible. But the reason why it's very important and really interesting, it's also about the context for the Adelie penguin. So here you have all the colonies around Antarctica, the breeding colonies, and what we see is that the population trends are contrasting across this region. So with decreases, for example, in the Western Antarctic Peninsula, or increases, for example, in the Ross Sea. And our current predictive abilities for these changes in population size of Adelie penguins are at large scales, so the scale of a region, thousands of kilometers, and long time scales, so several decades. We can kind of do it with a lot of uncertainty, but at shorter term, on shorter time scales, and for local population, it's really difficult. So there is really a use for a framework that can do that. And also an important point for this species is that it's really linked to sea ice, which is a defining element of the Southern Ocean ecosystem. All right, so now on to the plan of the presentation today. So the more focused question that we're going to try and answer is, how is environmental variation affecting the population trends and the population dynamic of Adelie penguin? And to do that, we're going to look at 3 different vital rates for which we will try to understand the links between all the components, between the environment and the population size. So these rates will be reproductive success, juvenile survival, and adult survival. And for each of those rates, we will try to build this kind of timeline, this kind of sequence, to understand, to pinpoint gap, and maybe to predict what will happen to population size. And also something that is going to come back throughout the presentation is the idea that we will be looking for general patterns. So by comparing data with other population around Antarctica to aim for generality, for understanding the mechanisms that may be true at the species scale. Before we go on to the results, there are a few information that you have to know about the Adelie penguin and its environment. And the main one is the seasonal cycle of Antarctica. So here you can see the seasonal cycle of sea ice with huge increases in winter and very rapid decreases at the onset of summer. That's what you can see here. And the breeding cycle, so the reproductive activities of the Adelie penguin, are based on that seasonal cycle of sea ice. So we won't go into too much detail, but basically the Adelie penguins, they do their reproduction only during summer, so when sea ice extent is the lowest. And a phase of the breeding cycle that is going to be of interest to us later on is the chick guard stage. And during this stage, you have always a parent present at the nest. It's right after hatching that guards the chick, and another parent that is at sea foraging for food to feed the chicks, the chick or the chicks. And yeah, another key idea, key metric that we'll have to keep in mind for the rest, and after that, that's it. It's called sea ice concentration. What is sea ice concentration? We may say SIC later on. It's a value that we can give to an area. So for example, this pixel here, and that defines how much, what's the percentage, the proportion of this area that is covered by sea ice. So it's a measure we use to quantify how much sea ice there is in an area. All right, so I just told you that Adelie penguins were associated with sea ice, but how does that manifest? So what we have proof that, or evidence that at large scale, as I was saying earlier, the population growth rate, so how much are the population increasing, decreasing, or being stable, is correlated with the winter sea ice. But what's underneath this correlation with the population growth rates are actually correlations that have been made between the penguin vital rates, so survival, reproductive success, things like that, and sea ice measures. And one thing that is really interesting, and a word that I really like, and an idea I really like, is that this association between the penguin vital rates and sea ice can be quite specific. So it can have particular spatiotemporal windows, and types of sea ice may have different impact. So that's what you can see here. If you consider the sea ice at different scale, it may not give the same results. And I think this idea is embodied by a term coined by Emerson and Southwell in 2022, which is penguin relevant sea ice. Well, it's a bit catchy, but it's just to convey the idea that different types of ice may have different effect on different vital rates, for example. All right, so now how do we measure that? How do we measure vital rates in penguins in the wild? So this is the setup we used for the thesis, this long-term monitoring setup in Adelie Land. So it's located south of Australia. And so it's an archipelago, the Point Geology archipelago. And on this archipelago, there is a breeding patch, a subcolony located here that is monitored with PID tags, so RFID tags, which are really small. And this is the same kind of tag you could put in a dog or a cat elsewhere. And so the subcolony we monitor comprises around 270 pairs. So it's quite a lot of individuals. And at the bottom of the colony here, you only see one. There is also one at the top. We have RFID way bridges. So every time the penguin that is PID tag crosses the bridge, it's in identification, but also we have data on its mass, on its body mass. All right. So now on to the first chapter, reproductive success. And the first question, the general question for this chapter is how foraging behavior links sea ice to reproductive success. So to try and decompose this relationship. So first, a bit of context. What do we know on Adelie penguins that is important for this? So Adelie penguins are central place foragers. So this is an important term in ecology, and it describes individuals or species that have to come back to the same place when they go foraging. So here, the Adelie penguins, they are constrained, they cannot use all the sea they want because they have the constraint of returning to their nest to feed their chicks. And the reproductive success of this species, we usually calculate it, we estimate it as the number of chicks per breeding pair. So we count the breeding pairs at the beginning of the season, and then we count the chicks at the end. And it usually varies between 0 and 1.5. And so this reproductive success, because of the central place foraging constraints, it is influenced by sea ice, by the amount of sea ice in the foraging area. And so by food availability that is linked to sea ice. And food availability, it can be decomposed in food abundance, but for this, we don't know a lot. But what we know about better is about accessibility. So in situation, So in situation like this, you have here the colony, you can pay attention to the scale. When there is no fast ice, so this type of solid ice, the penguins, they can access the open water freely. They don't have to walk over solid ice to go foraging. And in other situations, there is fast ice like that. They have to commute over this fast ice. And this we know it plays a big role in driving their reproductive success. And basically the idea is that when the sea ice is like that, when there is fast ice, their reproductive success is very low. They don't manage to breed when it's like that. But what do the climate prediction tell us? Well, there is a lot of uncertainty in the sea ice forecast and sea ice prediction, but from the one we have, so this is from a very recent preprint from this year, and I'd like you to pay attention only to this curve here in light blue. What this forecast is that in summer, the sea ice around Adeliland, this is for Adeliland, so quite relevant, is going to decline. So yeah, we know, and it's the sea ice, but also we have other evidence that it's mostly the fast ice that is going to decline. It's it's also a recent paper here. So the question becomes, now we know the impact of fast ice, but what we would like to know now is what is going to happen when there is no fast ice, if there is really less, not a lot of ice, or if there is a little bit more ice, but still no fast ice. So that's the first objective of this section, to know what happens when there is no fast ice. And the second objective is to try to do this decomposition that I was telling you about between the environment and the vital rate by using also the behavioral or physiological trait. So right, onto the methods. So for this, we used the long-term study at its full scale with the data between 2010 and 2024. And the metric, the foraging metric, behavioral trait we focused on is trip duration. Why did we choose this one? So trip duration is just the time it takes for the individual to exit the colony, to come back and to feed the chicks. So this is really accelerated. This is a 10 days period that you can see here. So no worries, they don't go this fast. And yeah, the reason also we use that trait is because it can be obtained with minimal invasiveness. So for example, for our birds, the one that we monitor, we implant them once in their life with a pit tag, and then we get a lot of information, such as this information. So for this in the team, it's really our basic information. And you can see very well in orange, you have the periods when the bird is on land, and in blue, the periods when it's at sea. And you can see very clearly the different breeding phases. So you have the incubation, the chick guard, and then the crèche. But today for this talk, we're going to focus only on the guard stage. And the reason for that is because it's during this stage that nest failure peaks, so that the most nests, they fail. So it's probably a very important phase. And the amount of foraging trips of data that we gathered for this phase is in excess of 23,000. So quite a lot of foraging trips. All right, now what happens? So the first question, if you remember, was what happens when there is no fast ice to reproductive success? So a direct relationship between the environment and the vital rate. So what we see here is that we find again this negative relationship that we knew about a bit already. But what we can also see, if you look only at the fast ice absence scenarios, is that the relationship is maintained. So that's quite an important result. And the side result of that is that when there is almost no sea ice in the foraging area here, the breeding success is actually very high. So the Adelie penguins, a bird that is associated with sea ice a lot, they are actually able to tolerate a near absence of sea ice in their foraging area during summer. There is really this aspect of periods that we have to keep in mind. All right, now if we try to decompose this, to break it down a little bit. So first, the relationship between the environment and the behavioral trait. We can see that when the sea ice concentration in the area increases, the duration of the foraging trips that you have here and here, the sea ice, it also increases. So a positive relationship that is also maintained when there is no fast ice. And now the last part of the decomposition, the relationship between the behavioral trait and the vital rate. Well, we also see this significant and in this case negative relationship where the reproductive success decreases as a guard stage trip duration increases. So here, what this means is that we have a fairly simple to obtain behavioral predictor of reproductive success, and that is quite interesting to us. But to make it even more interesting, what we were interested in doing was looking at it at the circumpolar scale around Antarctica. So lucky for us, that's what we were able to do, because there are a lot of polar programs, a lot of nations that also collect this type of data that we could put together and try to see what happened to this relationship between trip duration, so it's guard stage, but also a bit from the crash stage. It's not really perfect in that regard. But what we can see is that the relationship is broadly maintained, the relationship between productive success and trip duration. So it's an indicator. So if we look a bit at the overview, having this relationship validated at the circumpolar scale is really important because it increases our confidence in using this behavior demography linkage in the future. Because likely, I mean, in the future, the condition may be different, but the condition around Antarctica are already quite different. And so, yeah, for the overview, we showed also that all this relationship, all these links in between were maintained when there is no fast ice. And if we try and build the timeline to collapse, as we said we would do at the beginning, it's where the usefulness of the visualization aspect of the timeline takes all its importance. As you can really see very well here that when the sea ice increases, the trip duration increases and the breeding success decreases. So that illustrates the usefulness of the timeline to visualize. In this case, just to visualize and understand. All right, so what happens after the breeding season? Well, after the breeding season, when young penguins are produced, they go at sea. And after that, the vital rate that matters is their survival. So we call that the juvenile survival. And the question we ask here is what is the temporal and also spatial variability in juvenile survival of Adelie penguin, and what may be the drivers of this variability? So first, for juvenile survival, it's really an interesting vital rate because it's quite mysterious, actually. So for when Adelie penguins leave the colony for the first time, they may not come back for another 2 years. And we have very limited information on what they do during those 2 years. So actually, we only have 2 studies that put loggers or trackers on those birds, and they showed that they could move thousands of kilometers during this period. And this lack of knowledge on what happens to juvenile is quite contrasting with the importance that this vital rate may have on population dynamics, which is this part, if you remember, of the timeline. And we have evidence for that, solid evidence for that for other bird population, including seabirds. All right, so our objectives for Adelie Land, so here for Point Geology, were to quantify the temporal variability of juvenile survival, to try and find and discover and uncover its environmental drivers, and also to do this cross-population comparison with information we have from other colonies. So how do we measure survival using the setup that we presented earlier? So we use what is called capture-recapture data. Well, in this case, it's electronic, but it doesn't really matter. And how does capture-recapture data looks like? So it's when you go every year at the colony and you assess whether you detect an individual or not. When you don't detect it, you put a 0. When you detect it, you put a 1. So for example, this one is probably still alive, and this one is probably dead. Well, this information, we do it for a lot of individuals, so in this case, more than 3,000, and then we put it in in capture-recapture models, in this case, multi-stage models. We won't go into the details here. And the output, what's really interesting is the output of those models is annual survival rate, in this case, specific to the juveniles. And so we were able to obtain those estimates between 2007 and 2020. And what we can also do with this kind of models is to try and assess what are the statistical relationship between juvenile survival and a set of what we call environmental covariates. So in this case, sea ice and other environmental parameters that we suspected would have an effect on juvenile survival. And another aspect I really wanted to tell you about all this is the strength actually of our study design. So usually, in many systems, you have here the main study site. You would try and detect only the birds that come back to this study site. But what happens if there is a juvenile that fledges from here, but only come back and decides to breed in this colony, for example? Well, you may miss it and you may estimate survival not really accurately. So what we did, what was started in 2013, was that mobile RFID antennas were deployed across the archipelago. And this was really useful to control for a short distance dispersal. All right, so now onto the results. So we're going to see a time series of juvenile survival, and that is this one. And the first thing that we see looking at this time series is that there is a lot of interannual variation. So it varies a lot, but despite this variation, what we also observed is a 2.5 annual decline in juvenile survival, which equates between 2007 and 2020. This equates to a 32% decline in the survival of young penguins. And this is on top of that, on top of this decline of juvenile survival, you see that there is no data point here. It's not for no reason. It's just because on those years, there were extreme sea ice events during the breeding season, and no juveniles at all were produced in the entire archipelago. Maybe one in 1 year, but it's as if it was nothing. All right, so now we wondered what is responsible for this variability. So that's where the environmental covariates come into play. I'm sparing you the ones all the ones that we studied, but I'm just showing you the most significant, the most important ones. So what we found is a positive relationship between the sea ice concentration in April. So April is the moment right after the fledging, they leave the colony, and the juvenile survival. So that means when there is more sea ice in the area, in the vicinity of their natal colony, when they fledge, they survive better. I mean, that's the correlation we find. And this is also in good accordance with previous results showing that this post-fledging period is very critical to juvenile Adelie penguins. But we are not satisfied with just this general relationship. And if you remember the term penguin relevant sea ice, we wanted to understand it a bit more. And what happens is sometimes, again, pay attention to the scale, sometimes the environment in April around the colony looks like that, so quite dense sea ice with those areas of open water that are called polynyas. And in other years, it looks like this, so much less dense sea ice with a lot of open water in between the ice blocks. And what we found by looking at this in more details is that there was a positive relationship between the survival of juvenile and the amount of sea ice in the area that was heavy sea ice, so compacted, dense sea ice. And we found also the opposite relationship, that there was a negative relationship with the amount of loose sea ice, of fragmented sea ice. So that goes on to illustrate well, I think, the term penguin relevant sea ice. All right, now we've identified potential environmental drivers. A point we were also interested in: what are the demographic, what are the population dynamic consequences of this decline? Well, to that, we can gain insight from looking at what happened at the other colonies I told you about a little bit already. So the first study to report time series of juvenile survival was one made by Americans, Hink et al. in the Antarctic Peninsula, so here. And what they found is that juvenile survival at the study sign, it declined by 1.3% a year between 1982 and the year 2000. And the consequences for this, here you have population size and the same scale here. The population, it declined after that. And maybe if it was one population, that wouldn't be really significant. But what the Australians, Emerson and Southwell, observed a few years later, a time series that is a bit different here, they also observed a decline, and that was also visible in the population size. So what's happening at Point Geology with our decline in juvenile survival? Well, quite surprisingly, what we see for now is indeed a decline that is of a magnitude that is comparable, if not higher, than at the other places. But this is the population size. So the population size at Point Geology is not decreasing at all. And I'd just like to point out that the time series here stops in 2019. So, I mean, other people have the data, and that would be interesting to see what's happening now. All right. So how can we explain that there is no decline yet at Point Geology? Well, the first aspect, again, we will look at the other study sites. The first aspect that is really important to consider carefully is that the time series at other sites, and yeah, again, this is not the same time period exactly. Yeah? The time series at the other sites are longer. So maybe there needs to be a longer time period during which juvenile survival declines for it to generate an abundance decline. And also what we may understand, we may see from the Bécharvez Island, so the green one, the green population, what we may see happening is that there is actually kind of a threshold or a delay, between the moment juvenile survival declined. So here, 10 years of decline in juvenile survival, but the population kept increasing for 10 years. So that illustrates something that correlates well with the idea of the timeline to collapse, is that there may be delays in the moment a vital rate starts decreasing and the moment the population size starts also decreasing. So if we construct it here for juvenile survival, you see that the behavioral or the physiological link between the environment and the vital rate is lacking, and we will discuss about that more at the end. All right, now on to the last vital rate that we will examine today, adult survival. And the question here, the focus for this part will be more on the consequences of extreme events on this vital rate. And adult survival, yeah, I'm always a bit interested in this vital rate because the theoretical model, so in theory, what we know of adult survival in long-lived species is that it should theoretically have big impact on population dynamics. And a consequence of that is that it often shows low variability. So if you imagine here a time series of adult survival, you have adult survival in time here, usually it looks like that. So really stable, really flat. It doesn't vary much because it could affect the population dynamics too much. But what we know recently more and more from extreme events is that, yeah, sorry, something I forgot to mention is that when it's flat like that, we say that it's buffered. The theory that says that it should be flat and show low variability is called demographic buffering. So that's why we say it's buffered. But extreme events, so events that happen only that rarely happen, individuals may not see a lot of time in their life, they can overcome this buffering. So how does a time series of adult survival look when demographic buffering is overcame? Well, it looks like this. So it's mostly flat and then it drops. And the reason I'm telling you about this is because this kind of black swan events, that's how they called, we have a recent study from 2022 that says that they may have a key role in driving the population dynamics of Adelie penguin. And what's also important about this study is that this is a simulation study. So what we would be interested in is validating this simulation with real data. And so the reason we are able to ask this question about the impact of extreme events is because at Point Géologie, at our study site, we actually had 2 extreme events only in the 2010. So what were these extreme events? So these were extreme for the specific period, so for January, and usually it looks more or less like this, but in 2 years, 2014 and 2017, it looked like this. So a lot of extensive summer sea ice. So it's a specific type of extreme event that we are looking at. And what this extreme event caused were massive breeding failures. So in 1 year, the whole archipelago, so it's a population of around 40,000 breeding pairs, they produced no fledglings at all. And in the other one, they produced only 1. So 1 fledgling for 40,000 pairs, it's not a lot. And that's what we can see here. So 2 black swan events for reproductive success. But the question we asked is do these extreme events, do they affect also, on top of affecting reproductive success, do they also affect adult survival? And because adult survival is supposed to be buffered, how can it be that such events, they affect this rate, whereas it's supposed to be buffered? So the methods are really the same that we use for juveniles. It's capture-recapture analysis, a very similar model. And what we wanted also to do for this vital rate is to control for potential covariates that could explain variation in adult survival, because we don't want to have confounding effect of those covariates. And so we didn't select them blindly. We had other studies pointing towards some specific covariates. And we also used information on the movements of the adult birds in the winter season from other studies to select, if you remember the figure from the beginning, to select the most relevant spatiotemporal scale to measure the environmental covariates. So, all right, what did we find? So these are the 2 extreme events, and this is the adult survival time series. And what is striking right away when you look at this is that in 1 year, in 1 extreme event, so the one from 2014, adult survival didn't drop. It wasn't significantly different from the other years. But in the other extreme event year, so 2017, we can see a 15% drop in adult survival. So it may not seem like much, but for the adult survival of such a long-lived species, it's actually quite relevant. So only 1 black swan event for adult survival. All right, so what about other drivers I told you about that we wanted to control for? I'm only here showing you one, so the concentration, the amount of sea ice in the molting area, so where birds change their feathers. And you can see here, it's well illustrated that it cannot be those other drivers that generated the pattern in 2017. It sits really outside the distribution of the other survival estimates. All right, so how the second question we wanted to ask is how can this be? What can we can we infer from what we observed to get a bit more idea about the mechanisms? So this is the two contrasting years. And yeah, I told you that those years were both extreme, but actually when we look at them in more details, we realize that there are some differences between them. And notably, one of those important differences is that the access to open water was much shorter in 2014 than in 2017. So our hypothesis, our speculation about this is that in this year, when the birds, they failed the breeding, they stopped breeding and they wanted to go back to sea to feed, to replenish their energy reserves, they had to cross a distance that was too much for them, too much for the energy reserve they had left compared to 2014. So this represents what we believe is an immediate cost of attempting to breed in a year where it was this difficult to breed. All right, so now for the overview of the adult survival part. So as we just told, we gained insight on the how extreme event may disrupt the adaptation of long-lived birds to breeding. And also what's important, and is not shown here, is that we showed that extreme summer sea ice events, extreme ice events, sorry, in summer, they can affect the adult survival, but we have evidence from other colonies that there are drops in adult survival that are not linked to extreme summer sea ice events. So there may be a role for winter events. And as for the other sections, if we build the timeline here, well, we see that we also have a missing link, but this is ongoing. We'll talk about it right after. And we also see the limits of this framework, this timeline to collapse framework for predicting when the events, when the disturbances, the stressors, they are pulsed. They are not ramping stressors. And so for this, the framework is not really adapted when the vital rate is driven by events such as black swan events. All right, so now onto the synthesis. How do we bring all this together? How do we synthesize all this information and these detailed links? So we can first start with a retrospective analysis, and this means answering the question: is the point geology population, facing impeding changes or impeding declines? Well, to try and answer this, we can build the timeline for reproductive success, for juvenile survival, and for adult survival, and that ultimately affect the size of the population that we have from collaborators. And well, actually, for the visualization and understanding aspect, well, it's undeniable that we have gained more knowledge by identifying these links, more detailed for reproductive success and maybe less detailed for the survival probabilities. So that's for sure, because we're able to visualize. And we also, a really nice use of this framework is to be able to visualize the lags, as we discussed for juvenile survival by looking at the other populations. And what's also interesting of the idea of using this framework with several timelines, several vital rates, is that we may be able to see offsets. So for example, this has been shown in TIDS, I don't remember which, great TIDS, I think, in Europe, when you had one vital rate that was decreasing, but another one that was increasing at the same time. So overall, the population remains stable. So, but here, the main problem we face, and that was, I mean, we kind of knew about it at the beginning because this species is so long-lived, is that the time series length, the duration of our study may not be sufficient, even though collecting all this data for already almost 15 years takes huge amounts of money and of work. And also something that we may be closer to solve is that we would need this information on the behavioral or physiological components of the timeline to have better predicting abilities. All right, so that was for the prospective synthesis, but we can, the retrospective, sorry, but we can also do a prospective one. And this means the prospective synthesis, answering the question, we framed it this way: how many extreme events, such as the one we discussed, is the population able to withstand, able to manage? So yeah, this is an ongoing thing, an ongoing project that uses matrix projection model. So again, I won't go into the math details, but these are just simulation tools that use the data. So this is how they are constructed, and this is the data they use, and this is the data we obtained in the previous chapter. So if we put all of this in a model, we can simulate different extreme event scenarios. And in this case, we simulated two such scenarios. The first one, where the extreme events, that's the previous knowledge we had before this thesis, affects only reproduction. And the second scenario, where extreme events, that's the new knowledge we have, they can affect reproduction, but also survival. All right, so what do we find? So here you have the population growth rates, and basically when the line here goes below 1, it means that the population decline. And here we have the extreme event frequency, so 1 per decade, 2 per decade, and so on. And what we see is that in the first scenario, where extreme events affect only reproduction, the curve goes below 0 only after around 3 events per decade, whereas if extreme events affect both reproduction and survival, then the population starts declining as soon as the frequency of extreme event reaches 1.5 event per decade. All right, but this was super, I mean, to me, it's super interesting, but this was still very local, very focused on the point geology population. So what happens if we try and synthesize and scale up the synthesis at the scale of Antarctica, and we gather all the demographic, most of the demographic information that we can have around Antarctica, and we can present it like this. So this is maybe a bit not so natural to look at. It's the density distribution. So you just estimate, for example, adult survival, and you see how this distributes on a scale. So here you have 0% adult survival, here 100%. So for example, the way to read this is that Admiralty Base of this colony, the adult survival probabilities are usually lower than the ones at other sites. And we can do that also for the other vital rates, so reproductive success and juvenile survival. And what does it brings us to be able to compare those colonies, to compare those populations, is that we can pinpoint which vital rates are most influent. And this is a very important idea. And for example, here, the fact that we know that in the Western Antarctic Peninsula, so this region, the populations are really declining, but it suggests that adult survival may play a big role because it's really different from at the other study sites. And also to have information on different populations. If the same thing happens at different region of the same continent, the scales are really big here. Between these colonies, it's more than 3,000 kilometers to give you an idea of the scale. So if the same things happen at different locations, it gives us increased confidence and reduced uncertainty for species level forecast. All right, so now on to the perspective that we can have this information on the importance of vital rate by comparing population, just comparing the raw estimates like this. But something that I'd really like to focus my efforts on in the future is to apply some tools that are called life table response experiments, and they can allow to measure the contribution of different demographic rate for a population. And then, so for example, this is an example in Norway, and you have in blue here, it means that reproduction is the vital rate most driving population trends. And when it's in yellow like this, it means that adult survival is the vital rate most responsible for variation in population size. And so doing this, it can allow to pinpoint which vital rate are the most important. And this is really important for us to be able to pinpoint which timeline, which component of the timelines we have to work the most on. And so this is linked to the flexibility of the timeline. If we don't focus on the vital rate that is most important, maybe the timeline approach is not so useful. So you can use these ideas to progress in this direction. And yeah, the second to last perspective, as we were telling, is filling the gaps that we have here in the timeline. And a really promising things that we are looking at right now is to look at variables that we can extract from our automatic wave bridge monitoring. And so, for example, here you have the change in body mass of an individual throughout the breeding season. And one of the metric that we would really like to be able to have is how much is body mass. So when it exceeds the colony, when it's empty, how much is its body mass changes throughout the season. That is something we are actively working on and making good progress on. And finally, now we're almost done. I'd just like to mention a last perspective. Maybe it's the more romantic perspective. Maybe it's because I'm a bit romantic. It's about biparental care. So so for the timeline to collapse, we're really focused at the population scale, so at linking behavioral or physiological metric with vital rate metrics at the population scale. But you have other models, such as the integral projection models or IBM also, the other types of model, that use relationship between an individual trait and an individual measure of fitness. But the problem with biparental care species is that you have indeed this relationship. So the performance of the chick, the mass of the chick, the number of chicks is going to be influenced by the first partner, but it's also going to be influenced by the second partner. And this is something that we are looking at the moment. And also how, so for example, using the data on body mass that we just showed earlier, and how the body mass of the male partner and the female partner may be managed in tandem between those two. All right, and yeah, to conclude, just before the acknowledgements, I'd like to well, it's a bit of a catchy sentence, but just it's a bit of semantics maybe, because I used to mistake prediction and anticipation, but the idea here is we can have all the best prediction, all the best forecast we want. If we do nothing with it, and doing means anticipating, it doesn't really matter. And this decision of anticipating, doing something with the forecast doesn't depend on us. I mean, on us researchers, it depends on the politicians. All right, and so I'd like to thank you all for attending today, and especially the thesis jury, so Sandra, Richard, Jean-Yves, and Amélie. And also I hope you can notice the pun here, my supervisors, Céline and Nico. I'd really like to thank the French Polar Institute, that is the organism responsible for all the logistics in Antarctica. We couldn't collect this amazing data without them, especially the missions of the Terra Adélie 71, 72, and 73, with who I had the chance to work. And also all the partners and the lab I worked at during these amazing 4 years. So the Centre Scientifique de Monaco, obviously, thank you a lot, Denis and Sylvie. The Université de Moncton, actually we can say both because they speak both French and English. The IPHC in Strasbourg, the CEF Montpellier, of course, and also a lot of thanks to the Monaco Oceanographic Museum for welcoming us today. And also, oh yeah, oh sorry, I forgot about this. All of my amazing scientific collaborators, so Thierry Raclot and Jean-Pierre Desforges, especially, that were in the thesis follow-up committee, really super useful comments from them during those 4 years. And yeah, also all the co-authors and the collaborators, thanks a lot to you. And yeah, thank you. I think that's, yeah, that was just, yeah, I mean, do whatever you want with it. And then I prepared the slide for questions.
Theo, thank you very much. That was a fantastic talk. So I think we'll move on now to 30 minutes of questions from Sandra. Thank you.
Okay, I think maybe the floor is mine then to ask you some questions. Before I do so, I wanted to say congratulations. I found it an incredibly lovely thesis to read. I think you cited lots and lots of relevant literature. You must have been very busy reading. You've clearly done some exciting field work, amazing data set, thanks to your supervisors as well, of course. And I also found the thesis really beautifully illustrated, as we can also see here. So congratulations on a job really well done. And I thought, I mean, I have some nitty-gritty questions on some of the chapters too, but I thought maybe we can start out a little bit more generally. And I wanted to ask you how much you think there should be behavioral buffering towards reproductive success in your study species.
That is a very relevant, yeah, obviously it's a very relevant question, but and one I wondered a lot. So when we look at maybe I can go up in the slides. Yeah, okay, it's lagging a tiny bit. Where is it? It's here. Yeah, when we look at this, I mean, obviously you've read it in the thesis, but yeah. I was wondering, because when I first saw the data before fitting the curve, I thought, okay, and this is explained as is in the thesis that we did it a posteriori, that I was wondering whether there would be a plateau here, so an indicator of the foraging effort, the behavioral metric, increasing without effect on the reproductive success. Well, this is, as you can see here, it's maybe tough to believe from this figure, but when I try to fit a breakpoint regression to this, it indicated like a possible breakpoint around 30 hours of foraging trip duration. So yeah, I think it's possible. I think with less messy data, maybe that would show a bit more. Yeah, but so in penguins, that's also something we discussed a lot with the collaborators, because in ALCIDs, for example, this has been shown. I have maybe, oh, okay, yeah. I just prepared, because I think it's really important. I kind of knew that we were going to talk about this, because it's again a figure I put in the manuscript. But like the inspiration I took when I did that was looking at this figure on common years in the UK. And in this case, this is very clear, the behavioral buffer before the vital rate is affected. And we have also evidence, this is also a paper by Leighton Matthews, of this on the body mass of geese, well, not seabirds, but not so far. But yeah, in penguins, so I believe there may be something, because especially because I mean, why could they, in Adelie Land, they almost never, the mean foraging trip duration almost never, I mean, never drops below 16, 15 hours. In the Antarctic Peninsula, they are able to do trips in 9 hours, less than 10. So even though the buffer may not be as huge as the one the ALCIDs have, I think, yeah, penguins have some.
Yeah, so related to that, so you see that the trip duration responds to something, and then it is related to reproductive success. How about it being related to adult survival? So what do these long trips bring for adult survival?
Yeah, that's one of the metric we thought using, actually, because yeah, the behavioral or physiological component of the timeline is missing for adult survival, and the very metrics I really wanted to use at the beginning were metrics from the Weybridge system, because I thought they would be more informative than the metrics just from the duration of the trips. But as I couldn't crack, I mean, I think it's explained a bit in the thesis, I couldn't crack it far enough to have enough data to make this relevant. So, but yeah, that's something I'd like to use. But I'll be surprised. This actually links a bit to a work I did before the thesis with Kyle Elliott in Canada, working on pelagic cormorants on Middleton Island. And what we found is that the energy expenditure, either at the individual level or at the population level, wasn't related to survival cost. And I think the conclusion of that was that I thought was maybe it's just their condition, actually, that at the end of the breeding season that will determine their fate. And if we have only the measure of the effort they do, we may not be able to see the impact on survival. It really have to combine the effort with the condition it leaves them at the end of the breeding season.
And so would this also have to do with individual variation in quality behind your population level estimates?
Yeah, that's also the big question. Well, that's why I think also a strength, once we can crack the code of the mass tracker, a strength of this this setup is that we can have an idea of how low during their life individuals can go in terms of body mass. So this way we would have a mean to scale the condition of an individual compared to what's its usual condition, and that would be a way to account for possible differences, because it's really possible. I mean, the idea of quality is that, applied to this case, is that there may be individuals that are 4 kilos, both of them, and one goes at 3.5 survives, and one goes at 3.5 and dies because of this decrease. So, but we would be able to control for that by looking at the longer time series, I think the longer picture with all the mass variation across years.
So maybe moving in a slightly different direction, you looked at adult survival and juvenile survival, and I was wondering whether you think that there are different drivers for these. So obviously you looked at the penguin relevant sea ice measures, and I must admit I'm not very familiar with sea ice measures, so I got a little bit confused about the different ones that you have used and how they do or do not connect to each other.
Yeah, well, most of the metric used are sea ice concentration, so really an area, usually hundreds of kilometers, and it's the percentage of this area covered by sea ice. The thing is, I think the big question you're asking is whether there is spatial, to some extent, but mostly temporal autocorrelation in the sea ice. Because the time frames at which sea ice influences juvenile survival and adult survival may be quite different, actually. So they may be connected if the sea ice, for example, in April, is also important in the winter where we have more evidence that it's affecting adult survival. But yeah, for this, I don't know. I know it's yeah, a point that I have to investigate more. Is how much, because it also depends on the scale, right? The temporal and also spatial scale. This autocorrelation. So yeah, and yeah. So yeah, I think it's a complete answer. Or if you're not convinced, I can try and develop more.
Yeah, so I was also wondering about these lag effects. You mentioned them briefly, but for example, these 2014 and 17 cohorts, the juvenile survival you of course take into account across multiple years. So whether these juveniles did or did not experience these two massive events, was that something that you took into account? Do you think that would also only matter for the breeding adults, or do you think it would also affect the juvenile survival?
Yeah, that's yeah. I don't think it would affect the juvenile because let's take the first extreme event, 2014. So the juvenile is born in 2013, or let's say 2012. 2014, the year of the extreme event, should have been the year where they came back to the colony to visit the colony. But the juveniles not being attached to the breeding site, and especially the first year where they come back, they don't come back as early as the adults. They usually come back in January, so quite at the end of the reproductive season. So I think also because they are not attached to the site, they may not, like if maybe they perceive that they have to cross too much sea ice, they wouldn't take this risk as the adult would do. So I think it plays on the detectability of the juveniles, this extreme event, so they may not be able to come back to be detected by our antennas in those years, but that's why capture-recapture models are for. But I don't think it may affect them this much.
I was also wondering about the predictive power of these adults. So could they not see it coming in 2017 that they needed to cross this exceptionally long stretch of ice before getting to the water and give up their breeding attempts a little bit earlier? I mean, they ended up with 0 reproductive success anyway. Could they not have saved themselves in the process?
Yeah, intermittent breeding, but I wonder, I mean, for other long-lived species, it's like to skip breeding is a really common strategy, and we have some suggestions that they do that at the southernmost colonies in the Ross Sea, but in Adélie, looking at the data, I haven't tested it properly, but just looking at a lot of data, the breeding cycle we could see with the RFID, we can have it with all the years, they just don't seem to skip. Our mature birds, I never see them skipping, so I wonder, I mean, that's, I think, one of the suggestions that there is different level of skipping depending on the latitude. But yeah, I don't think in Adélie they have this adaptation of skipping reproduction. They have do it all the time.
But even without skipping, would they not notice during incubation, for example, that things weren't progressing as much as they were maybe expecting or hoping for, and therefore giving up during incubation already?
Okay, now I get your question. I think it really relates to the distance they had to walk. Because there has been a lot of work in physiology. So before doing this kind of life history stuff at Point Geology, they used to do, the researchers there, they did a lot of physiology work, and notably they worked on the how is it called in English? When they stop eating.
Fasting.
Yeah, fasting. They did a lot of work on fasting and on the phases of fasting. And so they could show that there is a trigger that is hormonal and also a trigger of behavior, of body condition that triggers reproductive abandonment. But actually this trigger, the threshold at which this is triggered, it's quite likely that it has been shaped by natural selection over a long period. And what I would suspect is that those extreme events, they must not have happened a lot of time in this evolutive history, so that the threshold at which reproductive abandonment occurs doesn't allow the individual to walk over maybe more than 50, 60 kilometers of sea ice before being able to replenish the reserves. That's what made me, I agree, maybe that wasn't super clear when I presented it, but that's the idea.
So could you bring these figures back up of the places that they needed to cross to get to the open water in these 2 years? Because the direction of travel seems to be different in these 2 years as well. So, you know, you've drawn the arrow to where the nearest open water is. Do they know this? Is this repeatable across years? Do they prefer to move in one direction or another? Is there any navigational work on penguins?
I must first apologize because these are not tracks. These are me who drawn the tracks to the nearest. I should have precised this more. This is the distance to the nearest open water. But that is a very relevant question, and the answer is I don't know if they prefer. Definitely, when you look at the GPS data, when there is no fast ice, for example, they have preferences. They often go to the east, for example. But when there is ice like that, I don't know if like petrels, they have the ability to sense through smell the nearest open water, but that is a good question. I think that the Japanese, they did a bit of experiment where they translocated penguins and measured how much time it took them and whether they traveled in a straight line to the colony. I think to some extent they must be able to do it, especially since when they come back for breeding at the beginning of before incubation, it's not unusual that there is let's say 60, 100 kilometers of fast ice, and when you are 100 kilometers away from the archipelago, you don't see it. So they must have some navigational tools that they can use to do that. So I would suppose that if they can do it in one way, probably they can do it in another one. But then it would be super interesting if only we had more data on this to look whether the individual with the best navigational abilities were also the one to escape, to be able to better escape this kind of trap. Yeah.
And I was also wondering, do you ever observe mortality? So if, for example, in 2017 they were actually succumbing to having to walk so far over the ice, would you be able to, I don't know, with drone flights or whatever, see dead penguins lying before getting to this water body to forage and replenish their energy?
That's a question I actually asked, because I wasn't there, unfortunately, in those years, and I asked to Thierry Raclot, which is a researcher in Strasbourg that was there, and he told me that he wouldn't see many dead individuals at the colonies, but what he suspected is that, I don't know how, if he observed that really, that a lot of them would fall into crevices on the fast ice. So I suppose that may also be a reason for the decreased survival in that year. But yeah, your question really highlights the fact that for adults, what would be ideal is to have an idea of if and where there is a peak in mortality, even not especially in extreme years, but in general. And this we don't really know. I mean, we suspect it's during the molt, because it's another energetically intense period right after reproduction, which is also intense, but we don't really know.
Are there any type of loggers that you can use to figure out how this is happening, where this is happening, and when?
Yeah, that's for the juveniles, the paper by Hink et al. In 2020, they deployed loggers and they looked when so Argos loggers that would send the data, and they looked what was the main period where the loggers stopped working, so indicative of probably the individual death. We could probably do something like that for adults, but the problem is that we would, you attach the logger on the feathers, and so you would attach them to post-breeding individuals that would only keep it for a month before they molt, and then you would have to attach them again. So a bit cumbersome, but maybe possible. And that's, at the moment, unfortunately, that's the only way I see to do it. Because I tried to talk about with people that did cruises in Antarctica to see where they would observe the adults in winter. And we have a bit of data that shows that they are associated with areas of heavy ice concentrations. But yeah, apart from that, to try and see where they die, I think it's quite difficult. But I'm sorry?
Yeah, we have to retrieve them. You were talking about.
Yeah, because we retrieve only the one that survived, so that's also the thing.
So maybe I'm now doing the chapters upside down, but I just wanted to ask you about this last chapter on the adult survival. whether you've also tried fitting a covariate for how much distance they would need to cross as an alternative to your sea ice measurement.
Yeah, that's right. I didn't do that, but I don't know if there are enough years with fast ice that they have to cross. But I mean, if your question was at the population level, I think there are not enough years. But one of the things I started working on, and then I stopped to write the thesis, is at the individual level, because, and I think that was also a question from you or from Richard. But from the RFID system, we can have the date at which the individual is last seen, and there is a bit of variability in this date. Or maybe I put it in the thesis, I don't remember. Okay, I put it. And yeah, so at the individual level, I would suspect that the individual that have, when they exceed the colony, that have the longest distance to travel, they would be the one showing lowest survival compared to the one that have a shorter distance, assuming that they all leave the colony at the same body condition relative to their abilities/quality.
I think I have a few more minutes, right? Okay, so maybe I'm going to go now back to chapter 1 and ask a few more detailed questions on mostly the statistics actually for that one. So maybe starting with the data selection actually. So first you say that you focus on this chick stage because this is where the main or reproductive success differences get made. But you haven't actually quantified it, I think, in your thesis. So how, in terms of percentages, what is this peak nest failure? Are we talking about 60%, 90%, or?
Yeah, I don't remember the numbers. It's definitely, I mean, the studies that I cite on this is quite old. I've read more recent studies comparing the survival between the crèche stage and the guard stage, and we can see very clearly that the survival, I think, in the crèche stage is in excess of 90%, so really high in crèche. But I think your question is more about incubation also.
Yeah, it's more about the pre-hatching stage, because that would be... So what we see in the terns, for example, is that if they get in trouble, the sooner they quit, the higher their individual survival to the next season is. So it really benefits to know when you are winning and when you aren't winning. So I would think, I mean, it comes back to one of the previous questions a little bit too. It's like, can't they predict it during incubation already? So, and if you are not taking that into account, are you bringing a bias to your data set? Are you missing an important part? So the percentage of nest failure would be relevant for me in that respect. Like if it's 10%, maybe it's not so important, but if it's 30 or 40%, maybe then that would be important to take into account. And is there any reason actually from your data to not be analyzing it and having stage of reproduction in as a fixed effect, as a class variable in your models?
Well, it's also that the foraging constraints in guard stage versus incubation are not really the same. They are much less constrained, I would say, in incubation because they don't have to bring food this much. I mean, in incubation, they are constrained only by their partner ability to keep, I forgot the term again, fasting. Whereas in the guard stage, they are constrained by the need of the chicks. But yeah, we could include it. Another reason, it's more of a practical reason, is usually we don't miss the trips with the systems, but in the incubation, when the system is set up too late in the season, so it's the overwinterers that stay there for the winter that put it in place, so at the end of September, beginning of October, and sometimes there is a bit of lag, and the birds are already in the colony when they set it up, so we miss a bit of information there. And so that was also a reason to focus on the guard stage, because it's the one for which we have the clearest information. But I agree with you, and actually that's something I try to discuss a bit, but also I think that these two stages may also be linked, so it's not a direct causal relationship that we see the relationship between guard stage trip duration and reproductive success, because we have also evidence from other colonies that in the guard stage, birds that have a lower body condition, that they may have inherited from constraining, for more constraints during incubation, they have longer foraging trips in guard stage. because of their lower body condition that they may have acquired in incubation. So this is what, the way I figured it is that, okay, growth stage is also integrative of this, so it may not be so bad.
Yeah, so maybe related to this is that in your models you wrote somewhere that you're not accounting for repeated measures for your foraging, for your trip duration, but your reproductive success data, of course, includes both the successful ones and the non-successful ones. So I was wondering whether you wouldn't want to actually include that as a fixed effect and take into account the repeated measures that you have. What was the reason for not doing it? You have the data, so why are you not taking that into account?
So your question would be to look at the relationship between trip duration and reproductive success, but also at the individual level.
Yeah, or to, at the very least, at the population level, take into account the fact that you will have more trips included for successful breeders than non-successful breeders, and to kind of correct for that.
Yeah, I agree with you that that would be, yeah, now you put it like this, that would be indeed something I should have done.
Well, you haven't submitted it yet, you can still do it.
Yeah, I will, thank you. And yeah, it's really, so the setup is super great, but something we try to do, I didn't talk much about this, it was a Gaël Bardon thesis. We can assess, we can measure or more like see the sex of an individual or its breeding success just looking at the RFID patterns. And what we tried to do with Gaël is to identify the date of the breeding failure by having ground truth data of birds we knew when they failed and RFID data, but it doesn't work actually. So that's a reason also I didn't use many data of failed breeders, because they had to be in the subset of nest that we were visually monitoring so that we had the nest failure date. And we cannot automatically yet predict it from the RFID patterns.
So maybe one last very general question from me. So you mentioned that, we now want to predict into the future, but also we want to act. And you've mentioned that in your thesis as well. And I was wondering whether you have anything specific on your mind, because this sea ice cycle seems a little bit ungraspable to me. So what can we actually do once we know what the consequences of it would be?
Yeah, well, yeah, as you say, for stopping the sea ice declines, there is not much to do. I mean, there is not much at our level, but the idea, I think it's a lot of times the same idea. What we can act on is reducing the amount, the diversity of stressors the population are exposed to. And in this case, one of the stressors that is actually increasing at the moment after a period of decrease is the fishery for krill in Antarctica. So the Soviets used to, and maybe other nations used to fish for krill in East Antarctica. They stopped after the collapse of the Soviet Union, and now it's increasing again. Also, the Chinese fleet is increasing. So that's one way we can act. But I agree with you that for the sea ice forecast, at least for the next decades, there is not much to do. But not much to do, but also, like, that's also the idea of looking at several populations. So I'm not going to go too far and say that we could translocate birds from populations that are kind of more adapted or more able to respond to low sea ice, but at least that gives us an idea of how they will be able to deal with it.
Thank you very much.
Thanks to you.
Thank you. So now over to me again. Yeah, well, I mean, just want to echo what Sandra said. It was an excellent thesis. It was a pleasure to read and a great talk. Okay, so I'm just also going to switch from general to specific questions, just depending on my whim. The first one, and I think you allude to this in the slide that you showed up at the end, how much do you think that, or how much evidence, convincing evidence is there that morphological traits should show a response to deteriorating environment ahead of a vital rate? I mean, I know there's the paper on albatrosses that apparently concludes that wing length has shown a response to environment, but something like wing length and body size, there's a whole load of other selection pressures that and also just pure physics and enzyme or kinetics that would suggest that wing length should be maintained, except when an animal is starving. So, and I would think that would also apply to most seabirds. So as you suggest, you might see responses in some of the vital rates before a morphological change. And I think that kind of brings me on to the question about how much do you think you can generalize across species, and particularly, I suppose, from the terrestrial to the marine environment and from more, R-selected to more K-selected species?
Indeed. I think it really depends on whether or not it brings us back to the first question of Sandra about the buffers, the behavioral buffers. And that is something I was thinking a lot for a possible postdoc, for example, is to try and meta-analyze if we could bring together this kind of relationship between let's say a behavioral more than physiological, because I agree with you, size is maybe too tricky for this. A behavioral trait and a vital rate, let's say a reproductive success, because it's the easier one. And if we could bring together data on this for many species and just to see whether a threshold, a nonlinear relationship exists, that would be a good indication that there can be a change in physiological/behavioral trait before the vital rate. And this echoes another meta-analysis from Curry, you likely know about it, but it just skipped this trait aspect, and it just linked the abundance of fish, or forage more generally, and reproductive success, and it showed that across several oceans, several populations, several species, there was this nonlinear relationship where the food abundance decreases for a little while before the reproductive success decreases. And I would guess that behind this, I wouldn't be surprised as if it was like on the common years, that it was the behavior that was compensating in the meantime. So I think the applicability of the timeline really depends on that, and also I try to highlight it, but on the how much ramping the stressors are, because if the stressors are pulsed, as in the case of extreme CI7s, then it's not adapted for this kind of disturbances.
Yeah, thank you, that's great. So I've got another question. I'll stick with the life histories. Why don't Adelie penguins skip breeding sometimes? Is it because they have no real idea what the rest of the season is going to be like and they are effectively pre-programmed? But if that's the case, why is that not the same situation elsewhere when they do skip?
Well, for skipped breeding to evolve, it has to be, I would say, given the different species in which it is present, I would say it cannot have appeared randomly, so there must be selective pressures for it to happen. So I would say in the case of Adelie penguins, because the sea ice can break up, maybe this is really tough to predict actually during the season when it's going to break up. So if they arrive, there is a lot of ice, they may, well, it's a bit of anthropomorphism, but they may assess the situation and say, okay, it's bad, but probably it's going to break up, and then end up in this situation. So yeah, more often than not, I suppose that it breaks up and frees. Well, that's.
I'm sure you're right. I was also along the same lines. Is there any evidence that they changed the timing of breeding, or is that also relatively fixed? Because that might suggest there was more flexibility.
They are actually quite flexible on their phenology, at least their arrival phenology. That's one of the things I looked at a bit at the beginning of the thesis, and we thought about writing a paper on that with Céline and Nico. You had 15 days advance in their phenology at Point Geologie. I think it was linked to this story of the iceberg that got stuck, and the sea ice concentration at the beginning of the breeding season was higher, so they had to cross more. And there is also, but it's the results from that, they're not, I mean, to me, they didn't seem very clear that they would gain any advantage at advancing their reproduction. There is a bit also of discussion regarding interspecific competition with the gentoos and the chin straps in the peninsula regarding arrival on nest site. But in other regions, I don't think it plays such a major role for them, the phenology, as it does for other species such as king penguins, for example.
Yeah, I'm sure you're right. But do they also does laying date vary? Maybe it doesn't matter too much when they arrive. It's whether or not laying date changes that's important.
Yeah, that's well, I don't remember, I'm quite sure I read about this, but what I can tell you from our data is that this delay, this advance that they would take on the arrival at the colony was completely offset by the laying date. The laying dates were almost always the same. So they do this buffering in between the arrival and the laying. So maybe, but this would be interesting to see how this translates into their breeding activities, because what it means if they have shorter periods is that they have less time to build the nest and less time to feed the pair bonds. It's not the right term, but to bond the pairs.
Yeah, sure. Thanks. So, Theo, obviously there's a very strong focus on sea ice here, and I'm not going to actually talk too much about that because there's going to be some more questions on that from Emily. I was just wondering about other indirect effects of sea ice. I mean, is there, and the obvious one is, so sea ice is mostly we're talking about, bottom-up control, but is there any difference in the way that the predators, that the skuas might respond to the sea ice situation that could be maybe exacerbating or ameliorating effects on the penguins, depending on whether the skuas fail, for example, and that sort of thing? And just essentially, what about predation and what role does that play?
Yeah, for the squaws, actually, I'm not sure. I know that they may have different strategy among individuals squad, that some forage at sea, some forage more on the penguin colonies. So maybe when there is a lot of ice, the squaws don't have access, so they shift their predation on penguins. I know there is a really nice paper on that by Lise Viola and Christophe Barbeau that I read, but I don't remember the conclusion. They modeled the dynamics of the two species together. Actually, Christophe did that in many papers. I don't remember the results, but the thing I can tell you about is not with squaw, another predator, the leopard seal, and in the Ross Sea, there is not much information about this, but they are, in Adelie Land, like we rarely see them, the leopard seal. There is one or two for 40,000 breeding pairs, it's not a lot. But what they found in the Ross Sea is that the predation risk is quite high, and especially when the sea ice makes it that they have to use specific areas where the seal can hide. So probably it can play a bit also, but it's tough to quantify that, especially if it's farther away from the colonies.
Yeah, thanks. Okay, so again, we'll stick with life histories. You made a really interesting comment, in your introduction, and you were talking about the difficulty of distinguishing between life history strategies from life history trajectories. You put it very elegantly, and even then within life history trajectories from effectively the distinguishing cause and consequence from intrinsic adult quality. And I couldn't agree more, but how do we really tell what are strategies to some extent within individuals, and how distinguish that from effectively individuals just reacting to their immediate circumstances and their recent breeding outcome?
Yeah, I scratch my head on that question a lot. And the only answer I came up with, I have no idea if it's valid, but I would frame it as a fitness equivalence. So if you don't have fitness equivalence, you have trajectories. If you have fitness equivalence between different trajectories, they become strategies. So I would really bring it to this idea of fitness equivalence, but it's not really practical to apply it in nature to actually see that. Yeah, that's but hopefully, one thing that I read about recently, that you may not going to like the answer, is that actually for population dynamics, it may not matter this much. I thought before reading more for writing the thesis, I thought this was potentially very important, and we have paper by Stéphanie Genevrier, for example, showing that it may be important, but recently I read another one on snakes, or actually reptile, I don't know which one, I don't remember which one, saying that they modeled different scenarios of individual heterogeneity in strategies, and the impact, or turtles, maybe it was turtles, the impact wasn't this big. But yeah, sorry, it's I slided a bit.
No, that's great. What have we got? Oh yeah, this is a wrong order, but anyway. So do you think obviously there's problems for Adelie penguins if there's changes to sea ice extent. And of course, you know, we were seabird ecologists, we consider across species comparisons. Do you think the future of some seabirds might be more secured by, you know, stepwise shifts in range, like the breeding range? So effectively that would be something akin to your black swan events, but that suddenly subantarctic species might start breeding in the Antarctic. And first of all, might that happen? And second of all, for Adelie penguins, is there anywhere else to go? Or effectively, for the Adelies and emperors, really, if they can't cope with these sea ice conditions, they really, that's it. You know, they're never going to, would they ever survive in a subantarctic environment, or is that really not not an option? Is everything basically become more polar, and then, you know, options, disappear?
So yeah, regarding your first question, I think definitely some species can maintain themselves and can benefit from a shift in distribution, especially, as you said, the subpolar species coming to more and more to polar areas. This may, just a little information I wanted to add, this may depend on whether the new habitat, the newly available habitat remains favorable all year long, if they are not migratory, of course. And this is important. I think this is also highlighted with Adelie penguins, that the environment at different timescale may have different effect. So for those species to benefit, they would have to have a favorable habitat all year round. And now on to the second question, they can actually go south still a little bit, at least I think for the 21st century, especially in the Ross Sea and the Weddell Sea. I think the Weddell Sea in the Ross Sea it's more, they have more papers by Ainley, for example, as early as 2010. I mean, it's not so early, but anyway. And in the Weddell Sea, what we are seeing now and realizing is that the Adelie population on the peninsula that are Weddell Seaside, they are doing great, actually. You have colonies that Céline sampled 2 years ago that are above 1 million breeding pairs. So it's quite huge. And the emperor penguins, then, yeah, as you say, we will never see them in the subantarctic, that's for sure, because no breeding platforms. But I think for them, the population, they may be able to use the Weddell Sea more and more as this sea become less icy. Because at the moment, the problem with the Weddell Sea is not that there is not enough ice, it's that there is too much. So yeah, but this will work obviously only for a time. And also the idea that is important is that it may likely not be able to host big enough population to ensure that they are not wiped out by such extreme events, for example.
Yeah, thank you. No, good points. Okay, and so back onto methodological ones. Did you consider using the other kind of IPM, so integrated population models, so you could effectively analyze your capture mark recapture data, but then inform that with the count data? Did you think about that, or?
Yeah, I thought about it, and it was kind of a trade-off, because it's, I mean, the way I see it, well, maybe I shouldn't say that, but the reason I was drawn to all this is not for the math, it's for the biology, the ecology. So when I look at these models, I think the outputs, what we can get from them is super interesting, but the math may be, I mean, they frighten me a little bit, but the reason is what usually the way I see it is that they are used when it's really difficult to estimate the vital rates, because you have sparse data, because your data isn't clear, maybe your recapture rates are very low. That's not the case for us. So we were already able to have quite a good idea on the vital rates we were interested in with the usual capture recapture models. And also something that I'd like to add is those models, they're great. But if you have more than one vital rate that is missing and you try to estimate it, and you also have papers saying, yeah, they do nonsense. They just like find the average value that maximizes the likelihood or whatever optimization function you're using, and yeah, here you have it. So I think it's super useful in some situations, but in ours where we were already able to have quite good vital rates data with the classical models, it's yeah, maybe not needed so much.
I think you're right. Great answer. I must tell all the people I know who work with integrated population models and remind them of exactly that. No, I do tend to agree. You know, in a data-rich situation, they're perhaps not necessary. Yes, go ahead.
But yeah, sorry, it's just that it's funny. I see Guy laughing because we talked about this a lot this week. Another thing I didn't tell much about is here we consider that population variation in size, they are driven by vital rates. But there is actually for population that are in what we call a transient state, so that are not at the stable age distribution, they may also be driven by change in the age structure. And I think this is one of also the big, something that makes the IPM, the integral, the integrated population model really useful. To be able to assess also this age structure that may have some influence on the dynamics.
Yeah, thanks. And so just a general comment. Are you certain there's no transponder loss?
I prepared a slide on this. No, that's my, yeah, but yeah. But we could test it actually, and that's also a discussion we had with the collaborators. I am sure Céline thinks about it every once in a while. No, we don't. That's unfortunate. I mean, sorry, I don't. Maybe other people know. But yeah, I quoted this website. There is a journal called RFID Journal. I was quite surprised. And apparently the kind of founder and editor of the journal said, no, I don't think they, so. I mean, it's not super hard evidence, but...
Yeah, I mean, I think one issue is the malfunction in the RFID tags. The other possibility is they're lost shortly after implantation. As well, I wonder whether that's more of an issue than people tend to suggest, which of course you could test it by injecting a couple of pit tags into the same animals. It wouldn't be that hard. Or unless do they interfere when you're going over the detector? If you have two RFID tags.
Yeah, that's bad. That it doesn't work. And we actually have the problem because the way we do the pit tagging of the cohort, so we go in the colony, we catch the penguin, implant the tag, release it basically, and we put a bit of green paint on the cheek. But sometimes when the first one we do in the season, we try to do it really condensed, but if there are storms and stuff like that, it's a bit delayed. We may, usually we check if it's not already implanted, but we have some birds that have two pit tags, and what happens when they cross the weight bridge is that sometimes it's one, sometimes it's the other, and sometimes it's not at all, because there are interferences between the radio signals. But I have, with Coralie, that's funnily enough that's here today, I have dissected juveniles that died after being implanted, not because of the implantation, just because of the storm or predation. And I mean, you could see that the pit tag was quite well in place. Well, it's worth what it's worth as information. But, so yeah, I would say we go, let's say 3 centimeters, the pit tag is deposited 3 centimeters below the hole, and also usually when the penguin is standing up, the hole is towards the ground, and the pit tag is towards the ground, and the hole is up, so the chances that it comes out are lower.
Yeah, no, that's good. Okay, so I might just follow up with if we've got time, yes. I'm just going to follow up on something that Sandra was asking about, which was about whether you have planned or thought of testing for the effects of trip duration in incubation. You could give good reasons why this focus on brood guard is important, but it might also reveal something about the importance of sea ice extent at that time of year. I mean, because of course some animals do fail at hatching, and you could also, if you could identify hatching birds that have failed to hatch or not, that you would still glean some useful information from repeating the analyses in incubation. If so, do you think that would that show anything different about sea ice? About the benefits of sea ice, or what the optimal sea ice conditions are if you were to repeat the analysis depending on incubation, analysing incubation foraging trips?
Yeah, I would say this really depends on how much does sea ice block success to predictive foraging rounds. And in the case of incubation, the site they go the most during this period, at least in Adélie Land and in other regions as well, is the shelf break. So the moment where the bathymetry really goes down. And because of the upwelling, this is a productive zone, so they can go there and target So it's the Antarctic krill that they find there, opposed to the crystal krill that they get in the ice. So yeah, apart from that, I don't think, maybe too extensive. Again, if it's really, really too extensive and they have to walk too much, it may lengthen the incubation trips, and then it may be worse for reproductive success, but yeah.
Actually, how much do you know about diet?
Yeah, that's
how much whether it might have changed or might change depending on environmental conditions?
How it might change this, I'm not. The paper on the sea ice trend, the summer sea ice trend, maybe we can put it again. I mean, for now it's a preprint, and they quantify the time of emergence of climate change signal. Oh, it's not looking good now, but also for this figure for sea ice, penguin, and fish. And I think I honestly don't remember the direction for fish and krill, but what they are seeing for Adelaide, I'm not sure, but what they are seeing in the Antarctic Peninsula is that the fish, the ice fish, the Pleurogramma, that is associated with sea ice, its distribution is contracting southward, and what they found in their analysis there is that the breeding success the chick mass, so probably an index of their survival, is correlated with the abundance of fish in the diet. This is also true in the Ross Sea. So yeah, if the ice fish decreases, probably it's not going to be good for them. But at the same time, you have colonies where the breeding success is still high, in excess of 1, and they feed at 99.9% on Antarctic krill. So the importance of fish may really depend on the colony, but it's really tricky. I would really like to know why some can handle 99% krill, and why some they need fish in their diet to be able to breed. That's I wonder.
Yeah, it's a good, tricky question. Okay, another generic question. Do you think it's better to be, if you're a chick, in a cohort that's large, and therefore, because there are potentially some benefits in termity or krill swarms, maybe improving foraging success, or a cohort that's small, and then there may be more krill to go around? Could it go in either direction, or do you think it might be a quadratic relationship, perhaps?
I don't think there is, at least for the point geology colony, I don't think there is yet density dependence with food availability. So the other part of the quadratic relationship. But then for the first one, that's what the Australians, so Louise Emerson and Southwell, they demonstrated for their colony, is that their juvenile survival rates, they were correlated to the cohort size, negatively correlated. So the juveniles, they survived less when the cohorts were smaller, and the way they discussed that were alley effects, as you mentioned. But is it due to social foraging? I really wonder, and I don't know. I'd like to go there in the post-fledging period, and just maybe the people that go around in the boats, they can see that. But I mean, personally, I have C6, so I haven't seen much when I went back with the Astrolab. But I don't know if there are aggregation of juveniles. I don't think there are many. So for this, I don't know. And maybe if I can add one point, so it could be for foraging, but it could also be for sheltering, the benefit of being together. And both, so we tested, the Australians, they tested a relationship between juvenile survival and the winter wind chill temperatures, and they found a statistical signal for this. So we thought, okay, we are going to test it also. And we also found, I mean, it's not so strong, but it's a statistical signal that juvenile survival may be positive when wind chill temperature, may be higher when wind chill temperatures are higher as well. So perhaps this is, I mean, this maybe they flock together in the winter to protect themselves from the cold. I don't know. I don't really believe it, but that could be a possibility.
Yeah, no, that's an interesting thought. That also makes me wonder whether, if you had a population that was declining, whether the decline might accelerate because of some type of alley effect. Could be size of the molting huddle of the adults, or potentially the juveniles, I suppose, on winter sea ice, but also predation effects. As the colonies fragment, there's more edge to center, and skewers might have a higher impact on reproductive success. I suppose that points to the idea that you might have tipping points in populations. Do you think, is that possible?
Oh, yeah, definitely, and especially related to what you mentioned. I think it depends also on how many predators there are, so that the rates of predation are quite different from one colony to the other, depending on the abundance of squaw. But yeah, this idea of fragmentation of the breeding patches, that of course the amount of edges of entries for the predators increases when the colony size decreases because there are more isolated patches. Yeah, I don't remember the authors, but there is a paper saying that we could try and assess the health, it's a big word, but the status of a population based on how fragmented the breeding patches are. And yeah, so that was not really the question, but about tipping points, yeah, I really think, because, tipping points are just kind of non-linear relationship, and this is, I try to highlight it and to find them if we could. So I really think, yeah, that there can be yeah, especially in colonial species.
So, okay, and then, a question about the relationship between fledging mass and survival. So you included, I think, mean fledging mass, and then you included the individual mass in the models. So does that mean, effectively, the latter is relative mass of the chicks relative to the mean, if the mean is already included in the model? And well, first of all, you answer that question, and then if you do, I've got a follow-up question.
So, yeah, so it's, because including the individual, and I think the result on the mean fledgling mass is the same whether you include individual body mass as an individual covariate or not. And yeah, the reason behind this is, I think, in Adeliland there is variability, obviously, within cohorts, but almost all the time, the cohorts, they can, on average, be at a mass high enough so that their general survival is not jeopardized by this parameter. Whereas when you look at the mean values in Adeliland, it's always, it's quite often in excess of 3.3, 3.5, 3.4 kilos. When you look at the fledging mass in the peninsula, it's 3 kilos, or even sometimes below 3 kilos.
Yeah, that's fair enough. But, might that be an argument then you should just include fledging mass? Because it's always going to be a benefit. It's important to be fat. It doesn't matter whether if you're fatter than the rest of your cohort, that's, that doesn't really matter. You still should really, it's the absolute mass that's important, not how fat how heavy you are relative to the rest. You could, it's possible that the simplified model with just absolute mass would also be fine, unless you really think there are differences in body size, then it would make sense.
But that's the metric we used, only body mass.
But you did include min mass though, I think, didn't you?
But the reason we included min mass is yeah, we did actually, and we found the effect only for at the individual level. It's this quadratic relationship, but we didn't find an effect of the mean cohort body mass that would be indicative, like a proxy of the feeding condition during the season.
So that makes sense. Yeah, fine. Okay, perhaps I would just exclude mean mass from the model at that point, if it's a model selection process. But it doesn't really matter.
Oh yeah, but because I didn't test more than two. I used in all models. They used individual body mass as an individual covariate, which is different from the annual mean body mass covariate. And because the annual mean body mass covariate had no effect, it wasn't included in the models that included two annual covariates, for example.
Okay, yeah, no, fine, that makes sense. Yeah, so a general question. Do you know how well the breeding success of the Adelie penguins, how well that corresponds with the breeding success of the other species at Point Geology? And do you think that might be revealing about what's a good and a poor year and so on?
Yeah, good point. Well, there is, for the first extreme event, there is also a paper by Christophe Barracho that showed that the consequences were for the entire community of seabirds. Maybe there were some species that managed to escape these consequences, the flying ones. But yeah, For the 2017, I don't remember, but I think even for the snow petrels and or the Cape petrels flying over such long distances, I mean, I think they feed closer, especially during chick rearing, they feed still closer to the colony than the amount of ice there was in these years. And the emperor penguins, they just have a different breeding window. So, but they are also affected a lot if the pollinia doesn't open early enough during their chick guard, notably.
Okay, thanks too. And then my last one is: are there going to be any more black swans, or will they all be white from now on, given the reduction in the kind of extremes of sea ice in the last few years? And there was a record low, and it almost looked like we were entering a new state. May all the swans be white from now on, or will we still have some years with extensive sea ice?
Good question. Well, the trend for the last almost 10 years looks quite worrying. And yeah, I'm afraid I'm not glaciologist enough to be able to predict that. But also I think even the glaciologist, we have one today here, maybe we will ask him afterwards. But one of the things I understand from this field is that it's really difficult to have accurate prediction about what's going to happen. And the big ensemble models, the big climate models that we can use for temperature or of the ocean or the air, they're not so good at predicting past sea ice trend, especially in Antarctica. So I don't think we can say with a lot of confidence that it's going to go in one or the other direction. But again, my understanding is not super advanced. I think it's enough for, I hope it's enough for biology, but yeah, let's see.
Okay, thank you very much. Over to Emily.
Thank you, Richard. And yeah, thank you, Theo. That was, I really enjoyed reading the thesis, same as my other colleagues. And I enjoyed your talk too. I like that we share the conviction that foraging is key, but it might not be all. But I also appreciated a lot your circumpolar approach. I might be challenging just like your framing of it. You were talking a lot about prediction at the beginning, but as you say at the end, you said like predicting is describing. I think you're more describing than predicting, and which is good. I think what we have, I mean, you took that example of the cod. I mean, yes, we understood, like we see that they collapsed, but now like they're not recovering, even though the stressors, like the overfishing was mostly gone for a while because the system has changed. So I think it's more important to like understand what the mechanisms may be. And if you want to go to, and I think, yeah, I think your talk was also a call, not that much for prediction, but for like long-term studies, because we need a long-term like data series. We need data series that are long enough to actually allow us to look at mechanisms and to bring not only demography, but also behavior and physiology, and like you did with behavior. Yeah, if you can go to your last slide that you had prepared for questions, yeah, please.
We haven't used it so much.
Yeah.
I'm happy, thank you.
And yeah, talking about mechanisms, what do you think is happening? So I also want to point that I really much appreciated that you moved past just sea ice concentration, that you tried to look at like, you know, penguin relevant sea ice. But even then, I think you could talk more about what you think are the mechanisms involved exactly. Like, why do you think Adélie penguins are so dependent on sea ice? What do you think is happening at those like 3 stages?
For the juveniles, you mean?
For the juveniles especially. I guess reproductive success, your hypothesis is that like it's fast ice, and it's how much they have to walk to get to the ocean in time to come back. It's pretty clear. What about juvenile survival, and what about adult survival?
Yeah, maybe if I can show you the slide where they are the satellite imagery. So what I'm about to say, I must say that for now it's speculative. So we wanted to deploy GLS on the juveniles with Céline to have more insights on this, but we couldn't. But anyways, so my speculations by looking at this is like an important aspect of sea ice and open water in Antarctica is that polynyas like this, or openings, they are supposed to be sites where food availability is predictable and predictably high usually. So I would say in situations like this, my hypothesis is that juveniles are better able to locate this area where food is abundant, and they can stay there and have better chances of surviving. Whereas in areas like this, food may be more sparse, more dispersed in the habitat, and they may have more difficulties to locate it.
Do you have any other hypotheses about the role of sea ice? For like, sea ice is not only, you're like very much focusing about sea ice as a habitat for prey. Do you think they could have another like significance for adelies than just habitat for prey?
Well, okay, now I think I see where you're going. The adults, of course, I mean, perhaps it's not this, but we know, I mean, you know very well that the adults, they can use the movements of sea ice with the Antarctic circumpolar or coastal currents in their movements. So they'd use it also as a moving platform, but also as a resting platform during the molt for the adults. For the juveniles, however, it could also be, but I'm not so sure that, yeah, maybe this is a lower quality. I mean, the resting platforms are of lower quality in an environment like this compared to an environment like this. But again, this is very speculative, and also another aspect of the sea ice.
It is sea ice in April?
I'm sorry?
What month is that image? What month are those images?
That's April.
Okay.
Yeah.
It looks very different in the Ross Sea. Like April is still like pretty low, I guess.
All right. Yeah, usually April, I think this is in the manuscript, it's the moment, at least in Adeliland, where sea ice concentration, they go up really rapidly, really steeply. And yeah, so the last point that we discussed, actually, I gave this talk a few times before on juvenile survival, and where I was asked, what about predation? Depending on how the sea ice looks like. But again, I don't like to go into this detail too much because, as I said, we have a bit of information on leopard seal predation on juvenile, on adults, during the breeding season. Like, we know crossing the ocean land barrier is risky for them, but we can only speculate for juveniles here because we don't, I mean, I haven't at any moment read information about this, about the predation in the winter season by leopard seal.
Yeah, there are like different concentrations of leopard seals. At least in the Ross Sea, we have a bit of info on that, and I do believe that, yeah, there. And also, what is amazing to me is that adult Adelie penguins spend very little time diving in the midst of winter, right? It's like 2 hours per day. So I do believe that sea ice is more than just habitat for prey. It's also like a resting habitat. It's to get out of the water and be safe from predation. And yeah, I think it is still very much unknown. So it is a tricky question. But
may I ask you a question about this? Because sure. I really wonder, it's reversed, but I really wonder how like a 500 meter square resting platform is different from a 20 kilometer square resting platform. I wonder if this really makes, that's why I didn't want to go too much in this direction, and in the manuscript I think I was quite careful on this, because I would suppose even like a patch of ice of 100 meters by 100 meters, how is that a problem for, maybe it is, but I wonder.
I think it is. I mean, at least for like molting Adelie penguins, we know they want that piece of ice to last for the 2 weeks that the molt is going to take. And also they like multi-year sea ice to molt, so that they're also it's like rough ice. I think there's a lot of dimensions to sea ice that we're not like yet able to measure and include in our analysis. But yeah, having a raft sea ice and ridges, pressure ridges, from which you can like get sheltered from the wind at a time of like energetical challenge is very important. And multi-year ice is what is predicted to be decreasing. So I think this is also something, but it is very difficult to have like data and long-term data on that.
Yeah, thanks a lot. That's also links up with juvenile survival and the wind chill thing. Yeah. What do you say? So yeah, okay, thank you. Thanks a lot.
Might be a thing, but. Yes, about the adult survival also, the fact that it's not maybe as buffered as we would expect for all the time. How long-lived do you consider the Adelie penguins to be?
I would say intermediate high on the tree of life. The generation time, I mean, it's in between 10 and 13 years, so it's still quite long. The maximum lifespan with the pit tags, we don't really know yet, but I suppose it's below 25 years. In our data sets, we have birds that were tagged as adults, as breeding adults, so at the very least 3 years old, that are now 20 years old, so they are at least 23, probably more. So yeah, obviously compared to maybe the extreme, that would be albatrosses that can live up to 80 years. Yeah, they're not super long-lived, but I would say intermediate high, in between intermediate and very long-lived.
I agree, but it's funny because we always like say, yeah, Adelie penguins, they're like long-lived species. Yeah, they're long-lived, but they're like less long-lived than most seabirds in the end. And they're also than a lot of seabirds, including like, I mean, Wilson storm petrels, how long do they live? It's crazy.
Yeah.
So, and why is that? Is that also because they're not top predators? They are not at the top of the food chain. So I think, again, predation, and again, it's something that is very like not studied enough because it's not easy. But I think it does play a big role in the life history of Adelie penguins. But that was super interesting to show also the buffer and the sea ice, and yeah. I think I had another question also about individual quality, going back to what Sandra was saying also. Yeah, I think by looking at the chick guard, you are biasing a little bit for like more successful breeders because they didn't fail during incubation. And also, you're also, I mean, we just have to like stay aware that we're looking at breeders already. We're like excluding all the non-breeders, which are even less successful. So it is something. And you had that figure 5 in your perspectives. I don't know if you have it here.
Oh, in the perspectives of the thesis?
Yes.
Of the manuscript?
Well, you were seeing the sea ice conditions when individuals were living early versus late. And you were like, oh yeah, individuals that live early, they're like less susceptible to be seen again because they're facing, your hypothesis was that because they're facing like high sea ice conditions, like lots of sea ice.
Is it
Right.
this
Yeah.
figure?
Yeah, this one. So can you tell me again what you were hypothesizing about that?
Also, so here you have the last detection at the colony, and here you have the density. So, because for this we would need the next step for that would need to measure would be to measure the distance between the colony and open water every day, where satellite imagery is available, then maybe fit a model to that to have interpolation, and to try and correlate the departure date to that. And just by looking at this relationship, it looks like the individual that disappeared, so the one in red here, they left the colony earlier than the one that were observed again. But the problem, why I didn't go too far on that either, it's also this problem of quality that comes in. It's because the ones that were able to withstand the tough condition the longer, so to leave the colony the later, are also supposedly the ones that are higher quality, maybe prime-aged, stuff like that.
Because, so why are those individuals leaving early?
Leaving early,
Yeah.
but I think they reached their critical energy reserve threshold.
So they're failed breeders.
Oh yeah, sorry, something I didn't mention. All of those birds are failed breeders.
Oh, they're all
even those
failed
ones.
breeders. Okay, that makes, okay, that's
Sorry,
totally different.
Actually, now I realize for me it was obvious that in 2017 there were no successful breeders,
Okay.
but yeah, I should have, thank you.
Okay, because for me it was like obvious, okay, the failed breeders leave earlier, they might be higher frailty individuals and, okay.
Okay,
They're
yeah,
all
sorry.
failed breeders, yeah, you need to look at that fast ice.
Yeah, thanks.
Awesome. Yeah, a little question. So I wasn't fully convinced when you say that the sea ice trip duration relationship is maintained between fast ice and no fast ice. But maybe you tasted that, you're going to tell me, but I feel like you should have fit the relationship separately. Because you're not, like sometimes your SIC range is not overlapping.
Yeah, I did fit it separately,
Okay.
and here I just showed it in an attempt to gain a bit of time. I showed only one figure, but
Okay.
I mean, that would have been smarter. I think you're right to show both of them and maybe to have the second one replacing the first one. But yeah, I tested it separately.
Yeah, thanks
But
for the clarification.
I think you're right to doubt because, I mean, if we count the blue points, it's not, we have to take it with a pinch of salt.
Sounds good. Last thing, yeah, you are calling for more data from the Rossi. I have a scoop for you, it's coming.
Yeah, Celine told me. Yeah.
There is a paper from Katie Duggar that is, and there is something surprising about the juvenile survival estimates. So your juvenile survival estimates, they're like 0.42 from pit-tag birds, and our estimates are from banded birds. And they're of 0.43 for Cape Royds and Cape Crozier, and 0.55 for Cape Bird.
Oh, wow.
How do you explain that difference?
Well, I would expect, I mean, we don't really have information on whether the effect on environmental condition and flipper bands is interactive or additive. But if we assume that it's additive, I would just expect that the juvenile survival is super high at Cape Bird. So I would say the real one could be 0.75, I'd say, because the papers from Duggar, they showed a 10% difference, which is...
Sometimes zero.
Also,
Sometimes zero, and sometimes, in Katie's paper in 2006,
yeah.
there is a difference of up to 10% in some years, and sometimes it's actually the banded birds that have slightly higher. I mean, it does vary with, it is not completely additive. There is an interaction, there is something going on that we don't know. And it was also on adults, and I do believe that the chicks that are banded from birth, from fledging, like probably react differently to bands than like birds that are like tagged as adults.
And plus there is, I'm sorry.
Yeah, still, yeah.
Plus there is, I mean, a bit related to the pit tag story, but there is a risk of band loss. I mean, I suppose it's stainless steel bands that you're using, but that they lose less than aluminum. But still there is a risk of band loss that is higher, I think, for flipper band than pit tags.
Yes, there should be, but yeah, we have a very small rate of loss. But we do have pit tags that fail. Like we had pit tags that fail. We had individuals that were like both banded and pit tagged, and that the pit tag stopped working. But we are not using the same pit tag, so it might be different. But yeah,
ours are good quality. No, I don't know. I have no idea.
They're huge.
True.
Duration. I think I have a few. Oh yeah, okay, last one for me. I was just a little surprised that you used data from the DDU weather station to calculate the average wind chill temperatures between April and September, but how far do you think the juveniles are from DDU at that time of year?
Yeah, 1,000 kilometers, I think.
Okay.
So that's really why I also do not discuss this relationship too much in the manuscript. And really the reason for its inclusion of this covariate is because the Australians did include it, and they did the same with their station in Antarctica. How spatially autocorrelated the wind chill temperature is in the bigger thousands of kilometers area is, I would suppose maybe a little bit, but it remains really speculative at this point. So yeah, you're right to point that. It's interesting.
That's good. Well, thank you.
Thanks to you. I'm looking forward to know what's happening to the juvenile survivor trend in the Ross Sea. Really.
Thank you. So I think next is Jean-Yves online, if you're around.
Yeah, thank you. Well, Theo, well done. Thanks again for inviting me, and sorry for not being with you today. I'm pretty glad. I saw you walking and warming up for a run, obviously, after the talk. I'm glad I'm sitting at home. Thanks for the talk was nice and nicely completing your thesis. I'm also glad I'm the fourth person talking. I had a few questions and few were already asked to you. But still I have some bits. For me, the most interesting thing, at least for my way of thinking, that when you talk about extreme events, where that's when there are lots of ice, when there is lots of ice around your colony and your study site. Well, usually when we talk about climate change, etc., people usually they think that actually the ice is showing, and so that was quite interesting. And I really like your way of investigating the different effects of the different types of ice on the different kind of animals. I mean, depending on the stage between juveniles and adults. That was great. But still, that means that the system is quite complex because the ice is not impacting the age classes the same ways, and I understand there are still quite a few bits to dig with this model, for example, for better assessing at the end, as you propose to do, to go from the knowledge to some decision making. So that was impressive. But there are still a few things that I couldn't really understand, and then I would like to ask you a few bits. Like just for example, on this slide that you have to shield the screens. On the slide you have on your computer that the reproductive success in relation to the sea ice concentration in the foraging area. It's not clear to me why 2013 and 2020 are absent fast ice, why they are on the side of the present fast ice, or what kind of ice is that in 2013 and 2020?
Oh yeah, okay, yeah, I thought you were asking about the other, yeah. Actually, what you can see here is that the sea ice concentration was high, obviously, but it was not from fast ice. It was only, at least in January, at the period of Chick Guard, it was only pack ice. Well, it was dense pack ice, but the penguins had access to open water straight away from outside the colony. Now I wouldn't believe that there is a very steep threshold at 70% sea ice concentration to explain the difference between the two. But yeah, to explain this specific variability, I wouldn't know how to explain it. I think if it's not the accessibility of the food, maybe it's the abundance, which we understand much less, especially in relation to summer sea ice. And also, yeah, a reason I didn't want I mean, I could give you the excuse of the reproductive success is also driven a lot by weather condition. So whether it's snowing at the moment of chick hatch, and whether there is a lot of meltwater in the colony that decreases reproductive success. But for 2020, there was not. So for 2013, I don't know, but for 2020, there was definitely not rain events in this season. Well, this is the case for 2023. The other points that is really falls outside the relationship. But yeah, probably, I don't know, maybe in 2013 is also closer to an event that occurred in the region, which is the grounding of an iceberg in the Commonwealth Bay area, which is around 100 kilometers to the east. To the east, yeah. So probably the condition also at the beginning of the breeding season were not so good. So that's what could explain why it's so low. But yeah, your question is more why it's so high in 2020.
Yeah.
Yeah, yeah, that's a fair point.
Okay, I really liked your approach for trying to to provide some general patterns by comparing the different colonies or populations, I don't know how to say. And then when you look, so in your manuscript, the next figure, that's about the reproductive success among, in relation to the foraging trip amongst colonies. It seems that at least for point geology, voila, thank you. So if I understand right, this figure, well, first I have a question: is why do you have more? Is your data set bigger for your study site on this figure compared to the previous one? That I didn't get the point. You indicate 18 sample size equal 18 for this graph, when you have 15 years of data. That's bizarre. But anyway.
No, I can. Oh, sorry. If you want, I can answer that.
Right.
Yeah, it's because, so the way I wanted to conceive this analysis was by using as much data as possible, and you have data on foraging trip duration and reproductive success for point geology that was collected before the RFID system was put in place. So I went and dig up that data, and if I may answer the previous question, because I thought about it in the meantime. The one I couldn't give you an answer to, why 2020 is so high on that figure. I think maybe it's not enough, but I think the reason I would invoke here is again penguin relevant sea ice, and for a same sea ice concentration value, I think as Amélie also pointed to it, the actual seascape may not be the same. So depending on the finer scale structure of sea ice, there may be difference that also influenced reproductive success.
Okay. So can you go back to the other figure? Is that correct? So based on this figure, is that correct? If someone conclude that point geology population is more sensitive or fragile in relation to the environmental conditions when it's translated in terms of trip duration? Because you have a relation between trip duration and ice condition, and then a consequence on the reproductive success.
Yeah, good point. I don't think it would be. I don't think we can answer that before we look at the relationship between trip duration and sea ice for other colonies as well. Because the thing is, if we look carefully, actually, the points that are driving these relationships are the ones from Bécharvez Island and Point Geologie, which are two colonies in the same ecoregion of, the same quite broad ecoregion of East Antarctica. And I think especially in the peninsula, they may not face such heavy sea ice concentration that they face in East Antarctica. Perhaps the exception to this would be Fukurokov, so the colony monitored by the Japanese also in East Antarctica, where it's actually a colony of only around 200 pairs, so really at the margin of the species breeding range. So you have basically almost no Adelie penguin breeding colonies before the peninsula west of Fukurokov. So rather, what you said, that they are more sensitive is one hypothesis, and the other hypothesis would be that they are not exposed to such constraining sea ice concentration at other colonies, as much as this data shows. Maybe they are exposed, but we don't see it in the years in which they were exposed are not, there is no data available for those years.
Okay, because I think that's something that was missing from my understanding, that actually some description of the sea ice context between the different colonies or areas. Because when I was looking in your phases, it seemed at least that the extent of the sea ice was actually narrower in Point Geologie compared to at least the western part of Antarctica. And And then I was wondering why. Do you know the processes through which the width of your ice cover, of the sea ice cover, is different between the different regions around the Antarctica continent? You get my point?
I'm not sure. I'm not sure what you mean by the width. So,
the extent. Sorry, the extent. The horizontal extent.
Well, it's definitely, so we're talking about breeding season sea ice extent. Even chick rearing sea ice extent, it's definitely much lower in the peninsula, for example. Okay. And really much lower. They have almost no sea ice in summer, at least on the northwestern part. And for the Ross Sea, I think it's a bit more tricky. I'm less sure about this answer, but I think the polynyas there is really extensive, and that there may be also, I think there is actually less ice in the Ross Sea, which is the rest of the year. This may be an ice-heavy sea, but at this moment, in the summer, there is less ice there than in East Antarctica. So I think when you look at maybe this is something I could have presented a bit more at the beginning, but when you look at how the global breeding population of Adelie penguin is spread across Antarctica, you have actually 1/3 of the birds, of all breeding birds that are, I mean, it's from data from 2014, so in the Ross Sea, so quite a small region, and you have 1/3 of the bird for the whole of East Antarctica. So the conditions there, well, it's not only sea ice, it's also the availability of breeding habitat, so apparent rocks, but it's not the perfect, I wouldn't consider it the perfect habitat for them, East Antarctica, contrary to the peninsula and the Ross Sea.
Okay, well, still you have a huge population. I mean, seems to be quite a big population, at least in point geology, no? At least compared to the two other populations where you have where you have the survival values?
Yeah, well, it comes now. It's the point is, what do we call a population, and that's a tricky point. Because on Bécharvez Island, it's actually what was really interesting. So the one where we have demographic data with the Australians. So they monitor the population on an island, but it's part of a bigger population. So it's as if in point geology we were working. I don't know very well their study site, but it's as if in point geology we were working on only one island. But what they're finding on abundance, I put them in the graph for just their island, but they show that it's the same for the entire meta population, so 150,000 breeding pairs. And in the peninsula, it's a bit the same. You have a lot of little, from what I understand, and I've never been there, but from what I understand, you have a lot of colonies / sub-colonies on little islands that are more or less demographically connected together. And yeah, this is a big, but just to give a definite answer, there is a nice paper by Santora from 2020 that tries and define what's a big colony, what's a small one, and point geology is an intermediate one, especially compared to the Ross Sea, where you can have colony of several hundreds of thousands of pairs.
Okay.
And recently, we also know that in the peninsula you have colonies of millions, 1 million.
Okay. So actually, I had a question about population genetics, but I think I would forget that.
No, that's a good. Yeah, there are.
When you compare your three populations, for which you have the juvenile survival, and you show that in point geology your survival is well, actually it's decreasing by 2.5%, which is much higher than the others. How do you deal with the fact that, beside the fact that the monitoring duration is different, but the period are also different between the different studies. So do you think that these different trends between the different say colonies are because of local pattern or because of temporal trends, more global temporal trends?
Yeah, well, it would be, at the beginning, I was tempted to think this, especially because the Antarctic Peninsula is the first region in Antarctica to show climate trends, so increasing temperatures, at least up until the 90s or the 2000s. But then I would have to reread the preprint on the emergence of climate signals in different regions, because I think that they look at this. But I would say it's more about, obviously, local drivers, so the amount of, let's say, the parameters we found that correlated with juvenile survival, which is sea ice concentration within 200 kilometers. It's local, so definitely a local parameter, but how much it is influenced by larger scale drivers, I don't know, and I'm not sure there is such a strong influence for now at the time scales we're considering, especially since we didn't find any evidence for links with, for example, large climatic oscillation, such as the Southern Oscillation Index, or which is the other one we looked at. Yeah, I don't remember, but those kind of large scales climatic variations.
Okay, so it's not a kind of illustration that maybe the climate change is accelerating during these 20, 30 years that you show with these three studies?
I mean, yeah, it's tempting to see it this way, but yeah. Because especially since in East Antarctica was actually showing an increase in sea ice concentrations up until recently, whereas the peninsula, it decreased much earlier. But yeah, I mean, I think, yeah, I wouldn't feel comfortable to frame it so starkly, is that the word? So strongly, maybe.
Okay. Okay. Just so that's a question that's a bit aside of what you did. That's more about the foraging behavior of these animals. You replied to many questions about polinias, etc. And you were talking about social foraging behavior. So do we know if Adelie penguins, are there any proofs or publications about social foraging behavior in Adelie penguins?
Not that I am aware of.
And do you know any species where that has been, any penguin species where that has been shown?
Yeah, jackass penguin, so Sphenicus, I don't remember the Demersus maybe, well, the one that is in South Africa, the African penguin, they do social foraging and they vocalize when they're at sea, so like possibly exchanging information. Okay. So, but the one thing to consider between those penguins and Adelie penguins and king and emperors is being a coastal foragers versus a pelagic forager. Well, Adelie penguin, they can also be coastal foragers, so to stay really close from the shore, but a lot of individuals and in a lot of situations they're also feeding more in pelagic areas. So maybe it may be less profitable to be a social for social foraging in such context. But that's a good point. And also maybe a hint we may have for this is that when you observe them departing from the colony for foraging trips, well, this may be related to dilution of the predation risk, but they often depart in groups and also come back to the colony in groups. So what's the part of this that is linked to social foraging versus risk avoidance or risk dilution for predation? I don't know. But I know people at the BAS, they are looking at this with the RFID detections, to see if the individuals that are nesting closer together in the colony, they also tend to depart the colonies together. But I don't know if there is an answer yet.
Okay. The cost of being the last one asking questions. You've been okay. So the setting in the study setting on plant geology, that's quite impressive. When you show all the different antennas you put everywhere for assessing at best the connections, I would say inner dispersal of your animal. If we consider all these, that's the same whatever colony or population. But still, I would say that's only 270 individuals out of your 35,000 individual of your population. How comfortable do you feel with the about 1% of the population that you are monitoring with this system?
Yeah, good point. And I think that's a question for a lot of monitoring program where it's actually impossible to monitor all the individuals. And so for survival, we could have other plots where we measure survival, but a good indicator of vital rate we can measure for many breeding plots and breeding patches, that's reproductive success. And there is a nice paper by Jan Meust, a collaborator that also works there, showing that the colony we monitor has the breeding success that is actually exactly the average of all the breeding patches within the archipelago. And when I saw that, I was quite reassured that this what we observe here may actually be how do you say that, be informative or relevant to the rest of the population. So yeah, the short answer is we have elements of comparison for reproductive success, but as you point out, we could very well imagine that survival, age-specific survival, is different elsewhere.
Okay, well, that's so prove that's a good setting and a good study model. I just have one final question. So you said you are romantic. I'm just wondering how philosophical you are as well. At the end of your talk, you mentioned that a huge amount of time and work for achieving this work. I know there has been decades of studies all around Antarctica, etc. That's also some cost of money and carbon footprint somehow. How comfortable do you feel in, and that's nothing personal, that's more have an idea of how you feel, and maybe also the jury, about spending all these energies period for actually providing knowledge and maybe advice for decision makers about a more global issue, which is overall biodiversity erosion and global change.
Yeah, well, I think what we can do, because also an interesting thing is that most of the what we call problems in biodiversity, we actually have the answer, the easy answer. Just the climate change that is driven by human activities. To stop it, or at least to make it flat, we just have to stop the kind of activities that generate that. So that's the easy answer. But what we do, I think, the way I see it, I don't do that to save the penguins. And I'm never thinking about this because that's completely irrelevant. But what I hope we can do is by gaining more knowledge, gaining more data, we are able to demonstrate more and more clearly how it works. And I think this is a way through which we can have a higher impact, is by how clearly we can demonstrate things. Because I really believe in the power, in the convincing power of a good demonstration. Like if you can frame something, if you have the scientific evidence, even though at the moment, scientific like the scientific method is being questioned a lot. But anyways, if you can really demonstrate something clearly and show that it's really well backed up by scientific evidence, I think it's a really good message. It's a really powerful message that we can give to the decision makers and to the rest of the population. And yeah, but I agree with you that we could say, and especially that's because in this PhD you could quite easily see the applied side of looking at these things in terms of conservation, even though ultimately what's the point of conserving Adelie penguins? But so yeah, the point I wanted to make is that for less applied things, so for example, if we were looking at I don't know, general mechanisms in ecology, you could wonder why would you go to Antarctica to do that? Perhaps you have a system elsewhere where you can do that, and I think that is a reflection we should have. So maybe if we're looking into things that do not necessitate to be done with the species or the systems that we have here, maybe we can start thinking to move them elsewhere. But it also hides a bit the fact that a lot of what we do is actually important. It's important for it to be there, and we can, on top of it, ask other more fundamental questions. And it doesn't cost much more to ask those questions there. That was a bit...
Oh, that's okay, thank you. I do tend to agree as well. Okay, well, merci, super, bravo.
Merci.
Thank you. That's great. So we've got 5 minutes from the two supervisors. So first of all, Nicolas.
Hi everyone, can you hear me now?
Yeah, we can hear you well, Nico.
Excellent. So, yeah, thank you, Joe, for the presentation. Very nice, as usual. We had also a good understanding of the amount of work that is required to understand something that is very complex, multiple scales, multiple years, and just looking at one species, let alone try to cope with many others, either from the food part or for the predator part. And I'm quite happy that there were many questions in predators today, and probably your ears are still ringing from me talking to you about predators in the field when we were the first time together. So it was interesting to see that. For me also, I don't know if I can ask questions a lot or not.
Yes, go ahead. You have your 5 minutes, now 4 minutes.
Oh, okay, time is ticking. So, yeah, I had a lot of questions over the years with Theo, and also Theo probably was some piece with that. But yeah, still some stuff that for me is quite crucial to understand for the future, and even with things that we were not able to have done in the time being, and that we know we want to work on. So I wanted to jump in on the aspect of whether we have enough data to try to predict what's going to happen for the populations, not only for point geology, but for the other ones. Are we in a time of crisis for Adelie penguins populations? Do we need 10 more years of data in some other places? What type of data? And it will be enough, and it will be too late.
So yeah, so your question, Nico, I'm sorry, it's just that I don't hear you so well, but just to sum up, your question is, I think I got the most. The question is what's the limit of how much data we need?
To try to come with a figure of what's going to happen for the populations, or just for DDU, and try to see that we can do something before it's too late, if it happens.
Well, in the case of doing something, since there are no many action levers we can use in Adelie at the moment, because there are, for the moment, almost no fisheries, I'm afraid the answer is there is not much we can do. But regarding how much data we would need, actually I think it's, for now, where we are at the broader time scale, where we are still in a phase where we try to understand things and to validate our predictive approaches,
I think, yeah, unfortunately, we need more data. Because when you think about it, it's a huge cost just to collect. I mean, it's not a just, but to collect this data is already a huge cost. But we haven't even covered the lifespan of the first penguin that were tagged. So in this first phase of understanding, of building our predictive models and of validating them, we need longer time series. But what I really believe, if we can keep this going, if these amazing long-term monitoring programs, they can keep running in a few decades, maybe in 2-3 decades, when we will have covered the lifespan of a few cohorts, of a few generations, then we will have perhaps enough data to be able to predict. But again, the nice but also not so nice aspect of that is that other mechanisms to be able, for us to be able to measure them, it may take more than decades. But I think to end on a positive note, I think with a few more decades, we will, I would say, have the bulk of the response that these species are able to produce in the face of variability. And also, I think we can have earlier confidence in our prediction if we look at information from other systems, other species. So, for example, I think the great and the blue tits in Europe that are also studied for a very long time and with a shorter generation time, they may be really informative for some things, obviously not for others, but yeah. I think that's...
Thank you. The other question, if the time is still good for this.
No, you only have 4 seconds.
Oh, okay, so, and there's no more round after this, right?
No, but I think with the indulgence of one of the other supervisors, I think, yes, go ahead and ask the question, please.
All right. For me, it's something that keeps bugging me for a while, and then you're quite aware of it, but try to understand this almost 50% decline of the survival for the adults in the time series. And why on earth a long-lived species, even if it's like a milligram long-lived species, why on earth they would commit to a breeding and put in danger their own survival, given like the trade-off that they usually do for breeding. And when you're long-lived, you try to avoid any cost to your survival. And so why on earth the decline happen? Is it something that is caused only by that year? Carryover effect? And from 2 years almost in a small time period between 2 extreme events? And do you think there's something more to this story?
I'm really sorry, Nico. I didn't grab all your question, but I think you're mentioning again one of your, I know, favorite subject as a shorebird biologist, which is carryover effects. And we discussed that a bit. It's true together a lot, and also with Céline. I really wonder for Adelie penguins. There is so much happening in between one reproductive event where we can actually observe the individual and the other one, especially in terms of energetic fluctuation. So they can almost lose 50% of their body mass during breeding, and they do that again during the molt in between breeding seasons. And I think for effects to carry over across the winter cycle, and especially across this kind of energetic bottleneck that is the molt, and that is mostly unobservable to us, for these effects to carry over to the next breeding season, I would be really surprised. But I mean, it happens definitely for other species, so why not for Adelie penguins? And I think that is, I think it was highlighted a bit. I'm not sure it answers fully your question, but that would be one of the reasons why looking at either the foraging effort during the breeding season or the body mass loss trajectories during the breeding season, what effect do they have on survival? And not only on survival, but also maybe on the individual level, I mean, phenology in the next year or the breeding success in the next year, so the other components of the life history of the bird.
Excellent. Thank you, Theo.
Thanks, Nico.
Thank you. Okay, over to Celine for the last 5 minutes of questions.
Thank you, Richard, and thank you, Theo, for this wonderful talk, as usual. Okay, so my comments and statement will be brief as a PhD supervisor. It will be more a general statement about the PhD, but still I will have a small question. Promise, Richard, I will be quick. It's often said that scientists seek for or to understand mechanisms that they do not yet understand, and especially mechanisms that they do not understand about themselves. Things that intrinsically raise questions for them about their own existence, their own behavior, and even some physiological reactions they experience. This is probably even more true for biologists, I imagine. And for you, Theo, of this PhD project, we saw that you explore various aspects of the life cycle. You spend a lot of time on juvenile survival, for instance. You spend a lot of time too on foraging trip, on how and where to forage, and how it impacts the breeding and the reproduction, breeding activities and the breeding success. But above all, and ultimately, and this strikes me actually, the common thread of your 4 years PhD project has been on the concept of timeline to collapse.
What was the end before timeline to collapse? I'm sorry.
The main thread
sorry.
your PhD was the concept of timeline to collapse. And this, I'm not really willing to comment on that, about timeline, about times, but maybe I will come back a little bit later about that. Instead of talking about timeline to collapse, if I take perspective of the nurturing parents like Nico and me, what matters for us actually during this PhD project and what drives us, I would say, during these 4 years of PhD project is a flip side. It was more the timeline to grow, the timeline to improve. And of course, there is still room for improvement during these 4 years of PhD. And I believe and I hope that Nico and me, we set layers of first stepping stones. But actually, there is no need for improvement on your side, I would say, when it comes to the abundance of ideas, of questions, and some of which at times take the form of truly existential inquiries, I guess, for you. And no need for improvement in critical thinking. I think this, you are quite, you prove again today that you are quite able to manage this critical thinking about your own work and about the work of the community, the scientific community. But on the other hand, there is still room for improvement when it comes to time. Time with a big T in French, in English, in Latin, and actually Theo is starting even with a T. And I think this was a sign. No, time and Theo, of course, is a whole poem in itself. Time and deadline remain a rather obscure concept for you, I guess. But in a way, I interpret your ongoing research question. I come back on what I was saying at the very beginning about timeline and time as the holy grail to be found and understood for your own existence. But well, I stop here with the comments, general statement, and I think you can understand everything here. I will just try to finish with a few questions, one question, actually. The question is, why not try applying the timeline of the timeline to collapse approach to human species. And in that case, how would you go about it? Do you have some idea about which parameters or which early warning signals would be interested to investigate for the human species?
That is a super interesting question, and thank you for the but it's funny, okay, I'm going to go completely freewheeling here, but I watched a documentary on Arte on the decadence of the Roman Empire, and I was thinking, because and there are also quite a lot of books, of very interesting books by, among others, an ornithologist, that was Jared Diamond, on the collapse of societies and stuff like that. And I find this super interesting, but at the same time, these kind of views, they also receive a lot of criticism. I know another book that was a bit alike, that is Sapiens, also received a lot of criticism. So I think definitely there may be things that we could see in the society and we would be tempted to see. Like, for example, I would say, but this is the point I'm trying to make is I find it more difficult to apply objective science. I'm sure sociologists can do it very well, and I really believe in the power of sociology. It's no question about that. But personally, I find it more difficult to be objective when it comes to, as science must be, when it comes to humans. For example, I would tend to think the early announcements of the decadence of a society would be people adhering to far-right ideas. But this is I know that I'm completely biased by my own convictions. But yeah, but maybe we can quantify that. Maybe the human species being such a social species, maybe we can try and quantify the diversity of the links that you have in a society, and that could be maybe a warning signal that something is about to go bad, whether the exchanges, the links are become stratified or things like this. I think this is really fertile ground for a crisis in human societies.
And would you like to investigate maybe different population of the species to maybe different areas of the planet?
Yeah, I think that's what anthropologists do quite nicely. But yeah, maybe one day I'll do that. Well. But before I have to solve all the Adelie Penguins questions, and I think I will have more than enough time to do that. So.
Okay, well, thank you. So I think, well, first of all, I'd just like to thank all the examiners and Theo for, and rapporteurs and supervisors for, there's been an excellent discussion. I think what happens now is the candidate and everyone else in the room leaves for just 5 minutes or so don't go too far, while the panel makes some final deliberations. So thank you. Oh, 20 minutes, sorry, 20 minutes. Okay, but actually, first of all, perhaps we could first thank Theo again for his amazing responses. Thank you.